grandfather back in the year 2020, the crazy, amazing year 2020. What a story. Never in my life have I had a year like 2020. And I've had some pretty exciting years, but boy, this year, it is really a crazy year. Hi, Kendall. So anyway, I have to say that, let me just double check things. I haven't posted the key to Quiz 7 yet, but it's ready to go. And I'm just giving, oh, sorry, Emily, you know what? Today the beard is just kind of free to do its thing today. Sometimes I wear, it's wild and free. Sometimes I bind it up like that. I mean, if I was to really brush it out, you'd see it's big. I try and control it a little bit, but sometimes it's hard to control. Anyway, hi, Travis. Anyway, what I was saying is that I haven't posted the Quiz 7 key yet. It's ready to go. It's possible that you don't want to try and take the exam until you've seen the Quiz 7 key. It's possible that that might be helpful for it. But I will post the Quiz 7 key as soon as class is over today. Give people a few minutes to act. Yeah, I will definitely post it today. But I got to tell you, a couple people have already turned in the exam, and I couldn't resist the urge to grade one, which I did. And that person got a 99 out of 100, though actually, when I was putting this exam together, I don't know. Now, believe me, I did it on purpose, but I put some extra points in it that are bonus points. So the exam's actually worth 107 points. 7 bonus points. Hey, you know what that is? That's 7% of the exam grade. So there's 7% in additional points. Well, anyway, this person I graded, I can't name names. It's against the law to name names. But this person got 99 out of 107. And I almost thought, wow, I would not necessarily have started this exam before I finished my material today, for sure. And but this person did very well. And I've also been grading on Quiz 7, which technically wasn't due until a little while ago, but I have graded a number of Quiz 7s too, so people may see their scores on those. I'm trying to get on top of it. What I am not very much on top of are the labs, but I'm going to get the labs graded and done. Look, we're not going to do any other labs. There simply isn't time to do any other labs now, and I apologize. I mean, I think when people buy into a virtual class like this, they're hoping for labs, and we did some labs, but we did not do as many labs as I wish we could have done. And you know what? Honestly, it's just me trying to figure out how to integrate all that stuff into the class and make it work and have it graded and receive all the stuff from it. So, we're basically not, I'm not going to post a new lab now. I mean, today is Thursday. We will meet on Monday to present material, and we will meet for a short period of time on Tuesday. But remember what I said on Tuesday was I wanted to meet for like a half hour as a group. And then I'm going to sit in front of my computer and have individual grade meetings with people. In case anybody wants to talk to me about grade stuff, they can do it during the normal class time. And so that's the plan. I mean, what we've got still have important material to cover. I know, I've been a student longer than anybody in this class, I'm positive. So I know that it gets a little questionable. You kind of have this whole incentive thing like, hey, why even bother to listen to this guy talk about connective tissue if it's not going to be on the exam? And the answer, I would say, is that, sometimes we need to learn stuff even if it's not going to be on the exam. So the material that we're going to cover today will be on the exam. I mean, it is material that is on the exam, no doubt about it. On Monday, that material, well, how can I put that material on the exam? The exam's already done. So on Monday, it's kind of like, hey, I mean, if you are so busy, you want to come and get this last bit of material on tissues, which will be useful to you in the future, for sure, be very useful for you in the future. Well, then, you know, I'm not going to try and force you. I'm not going to come to your home and knock on the door. So I understand that the whole incentive thing is maybe going to be undermined by the way I've structured things. But the truth is, I think that many people who are in the class are in it because they're engaging in a health-related profession's career path. They know as much as they possibly can in order to accept future cases and their career. So I'm saying that the Monday is going to be valuable. You're going to want to watch it too there, okay? So anyway, but today, believe me, we're going to be booming to a bunch of material. I apologize. So much boom through. Honestly, has it been a difference? Every day we've been booming material. Summer school. There might be a little less because you're 6 weeks instead of 5 weeks, but during a 5-week summer class, you have got a lot of material to cover every day. Oh, well, that's what we signed up for, right? So, and honestly, material can be successful on the exam. And in future, never want to forget the material that's presented today. You'll want to remember it and share it with your family. Okay? All right, so today is Thursday. It's the last Thursday we're ever going to meet. You're never going to need to look at me Thursday again. It's Thursday, July 30th. That reminds me of rent in the mail because it's due on the 1st. And today's 30th. So it's here. There's no cat next to me. Yesterday, I had a black cat. A big, it was Mouser. He was there on the table next to me for 2 hours of class, and then one hand on a mouse and one hand was a cat named Mouser. That's kind of blowing your mind right now. One hand was on a mouse, one hand was a Mouser. Anyway, it'd be a novel in that. Okay, so the work. We're gonna finish. People will be taking the exam. Exams due on Sunday at noon. Wait. I'm good. Well, thing to me, right? I'm the teacher, so let's look at the agenda. And is this the correct agenda for today? No. Well, it is, except the corrected dates. No. The dates. This is day 18. There's 20 days in the summer semester. So after today, we just have Monday and Tuesday of next week, and those are, well, Monday's going to be full scale blowout day, no doubt about it. Tuesday a little bit different. Okay, what was I going to say? So the exam 2 post due Saturday, August 5th by 11:30 p.m. So technically, it's not due until the very end of the day on Saturday. I mean, look, you can get it to me by midnight. You're doing fine. So that gives you plenty of time to work on the exam and do pretty well, I hope, right? Somebody pointed out that there is an error in the exam. It isn't a critical error. It's really just a free point, what it comes down to. So when I was writing this exam, I accidentally duplicated multiple. So there's 2 questions in a row dealing with mucus. If anybody has questions about mucus, different secretions, and exact same question, the exact same answer. But I'm not going to go and edit that or posted it. So hopefully you get it. Hopefully you get the first one right, and the second one you get it right too. So just make an extra point in there. Okay. And thank you for pointing me. Again, since I'd already posted the exam, I thought I could go back and try and pull that question and do this and do that and everything else. So stupid. No one's to blame for that but me. Of course, I'm the one accidentally duplicated, and I don't know how I did it. I have to figure that out. I'm not sure how I did it, though. I was booming on that exam in order to try and get it ready to go early day per student request. Okay, so the bottom line is if people love my exams. They always tell me they love exams. So anyway, it's ready to go, and it's due at by midnight, 11:03 p.m. Trying to figure out why I didn't just put it by midnight. Anyway, that means on Sunday I will be grading like crazy, so that by Monday people know the grades and start planning not in the final exam. I hope. Honestly, when I look at the grades that are posted today, I think probably very few people are going to want a final exam because people have really good grades, and that is just plain fine. Also, as it says here, quiz 7 is posted and due new today. The quiz 7 is technically already. If you did not get me, quiz 7, and I think people did get it to me. I mean, almost everybody got it to me on time. And again, I will post the key to it after class today for sure. I wanted to double check and make sure that people turned in. I'm going to say again that this text reading on tissues is well worth reading. I personally am not that huge of a fan of reading chapters in a textbook on the computer. I'm not. I'd rather have a textbook in front of me, but that's just not what we have. The truth of the matter is I used to have a chapter on tissue in the actual textbook, and it got to be really expensive for this one chapter, which had to be extracted from the textbook. And so this OER, Open Educational, is the future, meaning that many classes, at least in biology, I don't know what they're doing elsewhere, but in biology, there's a big push for us to provide free reading materials to people so they don't have to pay exorbitant prices for textbooks. And so this kind of reading a text that you have a link to like this, it is the wave of the future. There's no doubt about it. There's a big push to make these OER resources available to students. Okay. So today we're going to finish cytology 6, which was meiosis and genetic variation. We're going to move very quickly to PowerPoint, very basic embryology, and then we are talking tissues today for sure. Let me just look at this agenda and let me set myself up here. Oh, here we go. Oh, dynamite chats. I want to check this out. Ooh, is it breaking really big? How about right now? Yep, yes. Am I breaking up?
Kinda. I know there's like a little connection. Yeah. I mean, there's some points where we could hear you, but like, not really.
Could it be the beard thing? Do you think I didn't put the beard up? It's messing with the electronics? I doubt it. I mean, sometimes I get tempted to just go really slow, but yeah, the Wi-Fi stuck in my beard. Who knows? Anyway, the bottom line is that, well, we've got options. I'm trying to look. I can't see anything that I do differently. So if I'm breaking up badly, then maybe the thing to do would be to restart the class and give it a chance, huh? Do we restart the class?
I don't think that's going to do a whole lot. I think it's just overall probably your connection. I asked the original question because I want to make sure it wasn't on my end. So I don't know.
It's on every.
I don't think restarting is going to do a whole lot.
It probably won't. I mean, sometimes it does. Sometimes I have restarted a class, and when I restarted the class, it was fine. But you know what I've also noticed is that when I start a class at the beginning, it all seems fine in the first 10 minutes. And then something happens, and you believe me, I don't understand enough about you know the I don't understand about enough about activity to understand why they're at 12:30 than at 2:30 or something like that. I don't, but I imagine. I mean, I know that when it's lagging and I'm freezing, it's more annoying than it usually is when I'm not lagging and freezing. I mean, I'm annoying enough, I know. Then when you add that in, it even gets worse. So let's just plug away. Oh, there's no attachment link on D2L to submit the osmosis lab. Wow, I didn't know that. I'll make myself a note and see if I can correct that. Lots of people have been sending it to me by email, but I'll go back and double check on that. I don't want to do that now. It's only going to make things worse, I'm sure. So let's just plug away at it, and I hope that things that I said about the agenda are. Anybody have questions about the agenda, about due dates, about any of that kind of stuff? Okay. Then here's where I'm going to go. I'm going to plug away at it. Here's the PowerPoint that we were working on yesterday. And, well, I think I pretty much know where we were. I'm going to just double check my notes from yesterday. End of the class yesterday. I made a note. Before. We're just zooming through this. You could, of course, list one where I don't know people with their family life and I appreciate. Bottom line is that the most up-to-date version of this is posted on my email. No, what am I saying? The up-to-date version of this is posted on D2L. I posted it yesterday after it was updated. Okay. I even cleaned some of it up a little bit. There's where we were right here. This is slide 4, I hope. Oh my gosh. So look, we'll talk about some genetic traits in humans. And just take a look at the chart on the Oh, okay, good. Thanks. Where am I? Good. So I don't know what the
Yeah, I see you posted a picture about the ear. You ever seen a thing about the they're using your ear like a fingerprint, like a thumbprint to find out who you are?
No, I never seen anything about that. I believe it.
Yeah,
I mean, sure that
your earlobe.
I have a feeling that your earlobe is really distinct.
Yeah, like a computer can pick it out. And then the boy with the extra fingers, with the 6 fingers, they did something. If you watched the movie Gattaca, they made a person like that to play a piano. He could only play music because he had 6 fingers.
Remember, I saw Gattaca so long ago that I forgot many of the details about it. Well, so let's look at some of the genetic traits in humans just for fun. Look, notice here there's a column of dominant traits, like freckle dominant over widow's peak is dominant over straight earlobe. Free earlobe is dominant over attached earlobe. I mean, for what it's worth, dominant doesn't necessarily mean good, and recessive means bad by any. Okay. The traits in humans are dominant over other traits. It's worth kind of tracking this all the way back to the beginning of the story, which starts in the nucleus with DNA, and realizing that when you have traits like this, they are a reflection of your DNA programming. Remember that the relation that this was a quiz said, what is the central dogma of modern biology? How does the central dogma of modern biology explain the relationship between proteins and DNA? And the answer is straightforward. The recipe for proteins is found in DNA. These skin regions related to protein. Widow's peak, the function and the form of it related to proteins. Earlobes related to proteins. So recessive also doesn't mean rare. This is an interesting example. I mean, no freckles is recessive, but many people don't have freckles. This 5-finger, 6-finger thing is really interesting because there is a gene, the sexdactyly gene, that determines finger number, and 5 fingers is recessive. 6 fingers is dominant. So having 6 fingers is dominant over having 5 fingers. So you kind of want to say, well, wait a minute, how can that be? I don't know anybody with 6 fingers. But the answer, it's worth remembering that because something is dominant doesn't mean it's good or bad. Recessive doesn't mean it's bad or rare or common. And these things are not related. The bottom line is that, gosh, I can hear a bunch of background.
Oh, sorry.
He's got their microphone on and it's got a lot of background. Oh, thanks. So anyway, interesting. 5 fingers is recessive, 6 fingers is dominant. Why the heck don't we see more people with 6 fingers? And the answer is because even if it's a dominant allele, meaning it's a dominant form of the finger gene, it is just a very rare allele. It's not very common. Now, if you work in labor and delivery, you will see 6-fingered children once a month. And of course, when children are born like that and they have 6 fingers, the doctors or nurses will go to the parents and go, look, do you want us to just take that finger off right now? I mean, some places they just put a little rubber band around it and just starve it for nutrients, and it just kind of withers up and falls off. Other times they remove it. Later in life, you don't even know that you ever had 6 fingers. Your family's forgotten about it. And then the only way you might know it is if you have children and suddenly they have 6 fingers. So anyway, some genetic traits in humans. We're going to talk a little bit about genetic traits in humans today. That's where we're heading. You know, like the genetics of earlobes, just for the record, here's what it looks like, okay? Way this is inherited, the way that the genetics of earlobes is inherited is there's 2 genes, capital D, okay, which stands for detached earlobe, and it's lowercase d, which stands for attached earlobe. Now, what I noticed down here is the only way you're going to have an attached earlobe is if you have only little d's. That says recessive to me. You can have a detached earlobe if all you have is one detached earlobe gene. That says that the capital D for detached earlobe is dominant. Okay? So the detached earlobe, we know whether you inherited a key from mom, capital D, and dad. That's one possibility. Dad's sperm carried the capital D, mom's egg carried the capital D. Now you are capital D plus capital D. You are what they call homozygous, meaning you have the same 2 alleles, right? And you have a detached earlobe, of course. Now, because you're capital D, every sperm you make or every egg you make is going to carry the capital D. Now, it is also possible to have a detached earlobe if you have as one capital D, because capital D is dominant for detached. And as long as you get one of those, you're going to have detached. However, notice that there is D. Lowercase d is for attached earlobe, but it's dominated by the detached earlobe. You are then called heterozygous, which means you don't have the same two alleles. You have two different alleles in this case. Where did you get these genes? From your mom and dad. This is what is called the earlobe attachment gene, and everybody gets it, okay? But the question is, what do you get? Did you get one for detached and another one for detached, so therefore you're detached? Or did you get one for detached and one for attached, but since detached is dominant over it, you have detached earlobes? Thing is, though, is that when you have this genotype here, capital D, lowercase d, well, guess what? Half of your sperm, if you're a man, will carry the capital D, half will carry the lowercase d. And so there will be two different types of sperm. There'll be the capital D and the lowercase d. So the bottom line is you're only going to have an attached earlobe if, look at this, you get only the lowercase d's, meaning the attached earlobe. Remember, the gene is called the earlobe attachment gene, but there's two forms. Detached earlobe, which means that you can flick it around like, well, my earlobes are attached, so I can't. But if you flick it around, move it more freely, it's not attached to your head base or attached earlobes. So, this particular kind of genetics, we've got two alleles. One is dominant, one is recessive. And the relationship between them, if you get a capital D from mom and a capital D from dad, you're going to have detached earlobes. If you get a capital D from mom and a lowercase d from dad, you're going to have detached earlobes because the capital D is going to dominate over the lowercase d. There's only two alleles in this case. Capital D form of the gene and the lowercase d form of the gene. Okay. So is that the only way that we inherit alleles? No. There's actually cases where you have more than one dominant. That can be an interesting situation. Like it says here, when both alleles are the same, you are homozygous. When the alleles are different, you're heterozygous. In codominance, both alleles in the heterozygote are expressed at the same time. A heterozygote for sickle cell anemia has both normal and sickle cell hemoglobin. So take a look. Here's the father that does not have sickle cell, but carries the sickle cell allele. It's hidden and it's just not very visible because they don't have any physical effects or minimal physical effects from somebody's trying to get in. Okay, I'm back to here. So again, here's dad. He has very minimal sickle cell effects. Okay. Here's mom. She has very minimal sickle cell effects. Both of these are carriers. Do you see that? They both are heterozygous. The normal hemoglobin allele R and the abnormal hemoglobin allele lowercase r. Both do. Now, we call them carriers because even though they have no idea probably that they carry the sickle cell allele, nevertheless, it's hidden in their genotype. Now, when this man makes sperm, half of his sperm are going to carry the capital R and half are going to carry the lowercase r. When this woman makes eggs, half are going to carry the capital R and half of her eggs are going to carry the lowercase r. Now, look, if a capital R sperm fertilizes a capital R egg, then you get a person who's capital R, and they are completely unaffected by sickle cell. And in fact, they are not a carrier. They are no longer, that person will not be a carrier. Now, if a lowercase r sperm fertilizes capital R, or if an uppercase sperm fertilizes lowercase egg, then you get these heterozygous examples, right? Where you've got little r, capital R, little r. These are the same. But these are carriers. And may have their some cell. They might experience some sickle cell disease, but they might not experience much sickle cell disease. If a sperm carrying the sickle cell allele fertilizes an egg carrying the sickle cell allele, then take a look. Now we have sickle cell means this person has full-blown sickle cell disease. I mean, they got the sickle cell allele from mom and the sickle cell allele from dad, and they have sickle blood cells and lots of them. And they definitely have physical effects that will make them wish that they did not have sickle cell, for sure. You know, no doubt about it. So again, when it comes to this kind of thing, let's look at a real example of codominance, because human blood types are an example of that. I mean, like it says here, for many traits, there are more than 2 alleles existing in the population. I mean, when I think of things like the alleles that create height or create hair color or eye color or skin color, these are not determined by just one gene with allele. These are probably determined by genes with multiple alleles. Okay, but the thing is you never get more than alleles. You get one from mom and one from dad. The human blood group ABO is an example. There's 3 alleles, right? We call it the ABO blood type gene, right? ABO blood type gene. 3 forms of it: the A form, the B form, and the O form. So there's 3 different alleles. Now remember, you're only gonna get 2 of these. You could get an A from mom and a B from dad, an A from mom and an A from dad. You could get an A from mom and an A from dad. You could get a B from mom and a B from dad. You could get an O from mom and an O from dad, but you could get an O from mom and a B from dad. You can get an O from mom. Sorry, I got this brought back. Well, anyway, you're going to do these depending upon what your parents have. Again, they only have 2. So it's not like your parents can give you A, B, and O. One of them can give you A, B, and O. They can't. They only have 2 forms of it. But there are 3 forms of this allele. Now, like it says here, A and B are dominant over O, while A and B are co-dominant to each other. Meaning, if you get an A from mom and an O from dad, you're not even going to know you have that O. The A is going to dominate. You're going to be blood type A. But if you get, say, an A from mom and a B from dad, well, both of these are dominant. And really, dominant means, it sort of means assertive, and it will be expressed. A dominant allele is an allele that will be expressed regardless of what else is paired up with it. It will be expressed.
I have a question.
You get an A. Yes, go ahead.
How does that, like, A, B positive or B positive works? Because I know if, like, I had a friend, she was B negative and her son was born with B positive or something like that, and the body had to give her some special medication so she could keep the baby.
Oh, yeah.
How is that expressed?
Well, okay, so here's the thing. Right now, what we're talking about is one blood type gene called the ABO blood type gene. Okay, so but it's true. If somebody asks me my blood type, I just immediately say O positive. But what I'm telling them then is, look, my ABO blood type is O, but positive refers to another blood typing system. Okay, and that other blood typing system, I'll put it in the chat here. It is called the rhesus factor, right? It's often just written, they something call it RH. And it has two alleles to the gene, okay? It has a positive and negative alleles. So that's what the rhesus factor is. It has two alleles, positive and negative. Positive is dominant, and negative is recessive. So here's the possible combinations. You could be positive, positive. You could be positive, negative. Here's the three possibilities. Positive, 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 negative. Three possible combinations. So, positive, positive, negative, negative. If you're positive, positive, then you're going to be RH positive. If you're positive, negative, oh, well, positive is dominant over negative, so you're going to be RH positive. If you're negative, negative, well, you can guess. Negative is recessive. The only way you're going to see it is when it is homozygous for the recessive allele in this case. So we're talking about negative, negative. That means you're going to be negative. All right. So to go back to your question, though, it sounds to me as if in the case that you described, Kendall, the man was positive and the woman was negative, right? Meaning that the man, he was positive in the RH factor, and the woman that he impregnated was negative. And because he was positive, he at least half of his sperm, than 100% of his sperm, but at least 50% of his sperm carried the positive allele. And when fertilization happened, created a positive embryo, which was destined then to grow in the woman who was negative. Now, her body could see that positive allele, meaning they could see that unusual protein being produced in the baby, and as a result, it could actually start producing antibodies against it. And normally in the first pregnancy, it takes a while for that to actually happen. The first baby's usually not the one that's affected by any of those antibodies. It's the second pregnancy where the antibodies are already there thanks to your immune system. You get pregnant again, and now you're a negative woman and you have a positive baby in you, and now you've got antibodies already, and that can actually attack baby's bloodstream and destroy red blood cells. So it's a big deal. Again, I remember when I was growing up hearing that, oh, before you could get married, you take a blood test. And we thought, well, their blood herb or whatever. Gonorrhea or something. But no, what they're actually testing you for when they do a blood test before marriage, I don't even know if they still require that, is they're testing you for RH factor, because they want to tell you, hey, wait a minute, here's a circumstance where mom's positive and dad's positive, no problem. Dad's negative and mom's positive, no problem. Um, well, let's put it this way. They could both be positive, but they could still produce a negative baby because, remember, there's always two alleles. But the bottom line is that that they were looking for a situation where mom was negative and dad was positive, and so therefore there was a possibility they could produce an embryo that would be positive inside of a negative woman's uterus, and that would be a big problem. Right? So these days there's medications. I mean, there's antibody depressors that can work and solve these kind of problems or help with these kind of problems, so that you don't end up with a baby that has blood damage because it ends up being positive inside of a negative woman for that. So I think I said this early in the semester. I said, look, I think there's like 26 different blood types, believe it or not. There's ABO, there's rhesus, there's M, there's like 26 different blood types. Most of the time we don't even care about these other blood types because they are they're not super important unless you are going to have, say, an organ transplant. You want to be sure that you match up as many of those as you can. So in human blood types, just as an example of this kind of inheritance, there's A-type blood makes type A antigen. B-type blood makes a type B antigen. Type O blood makes no antigen. These antigens are polysaccharide. Excuse me. These are what's the word that I'm looking for? Sorry. These are combinations of, they're glycoproteins. Sorry, that's the word I was looking for. Glycoproteins that are attached on the surface. So there's this blood A that has this blood type A label, blood type B that has this blood type B label, blood type O that has no label at all. So the genotype is just the alleles you get. So you could get A from mom, A from dad, B from mom, B from dad, O from mom, O from dad, A from mom, B from dad, A from mom, O from dad, B from dad, O from mom. You can get any of these combinations, okay? These are the possible combinations that you could get in a hypothetical sense because there are three alleles, right? Remember in attached globe, it was really only three possible chances, right? You could either have two detached alleles, one detached allele and one attached allele, or two attached alleles. That's all you could get, three. But because there's three alleles here, we get six possible combinations. Now, what phenotype would each of these genotypes have? Phenotype means the actual physical measurement that you're going to get. So look, obviously, if you have an A from mom and an A from dad, you're going to be A. B from mom and a B from dad, you'll be B. O from mom and an O from dad, you'll be O. A from mom and a B from dad, well, this is codominance, right? A and B are both dominant. So you're going to be AB. A from mom and an O from dad, well, A is dominant over O, so you're going to be A. B from mom and an O from dad, well, B is dominant over O, so you're going to be B. Oh, like it says, you can be blood type A if you get either of these two combinations. You'd be blood type B if you get either of these two combinations. You'd be blood type B only if you get A from mom, B from dad, and vice versa. And you'll be blood type O only if you get O from mom and O from dad, right? So this is the physical body expression. Blood type A, blood type B, blood type AB, blood type O. Here's the genotype, meaning the underlying alleles. Now, just for the record, when this person makes sperm or eggs, every sperm's going to carry an A. Every egg's going to carry an A. That's all you have are A's to give. But this person, they make sperm or eggs, half of their gametes are going to carry the A and half are going to carry the O. So here's a question, believe it or not. I read this, on a genetics site, and it said, look, a woman has type A blood, and she claims a man whose blood type is B is the father of her child, but the child has type O blood. Is it possible that he could be the father? I mean, she's type A blood, he's type B blood. The child has type O blood. And, when you look at this, you think, ooh, wow, this is kind of an interesting conundrum. But you know what I got to tell you? This is exactly what's happened in my family. My mom is blood type A. My dad, blood type B. Me, Steve, I'm blood type O. And I'm going to tell you, no doubt about it, when I was born and they typed my blood, there were people in the family who went, wait a minute, you're A, you B, how do you have a blood type O child? And I think that they cast some minor aspersions on my mom. But a doctor who knew something about this was probably pre-genetic counselors, right? I mean, when I was born in 1955, I don't think they were paying anybody to be a genetic counselor. They just barely understood the genetics of some of this stuff. Though it's Mendelian, so we could take it all the way back to Gregor Mendel, right? Anyway, somebody said, no, that is entirely possible that you could have a kid with blood type O. Now, one of my blood type A, one of my brothers blood type B. Is this possible? And honestly, don't think of anything bad about my mom and the mailman or something, because I'm going to show you how this is totally possible. Okay, here's the thing. Sorry, this is sort of boring to look at, but let's consider this. My dad B and my mom was A. Well, here's one way my dad could be B. He could be BB, and if he was BB, every sperm he made would carry the B. Every child he had would be B in one respect or another. My mom, if she was A, but her actual genotype was AA, then every egg she made would carry the A. Every sperm carried B, every egg carried A, B fertilizes A, AB, AB, A, 100% of BAB. So clearly they're not this way, okay? Oh, my dad was BB, but my mom was AO. She would still be A, of course. The O is hidden. Dad would still be B. 100% of his sperm, would carry the B. But my mother, half of her eggs would carry the A and half would carry the O. So B fertilizes A, B. B fertilizes A. B fertilizes O, then you've got BO, which is B. B fertilizes O, BO, which is B. In this scenario, the only way their children could work out would be 50% AB, 50% B. Now, that isn't what happened. And so this can't be true either. The only way that, and this the way that's true, is my mom is heterozygous. She has AO. My dad is heterozygous. He has BO. And this B sperm fertilizes A egg, you get an AB child. B sperm fertilizes O egg, and you get a B child. O sperm fertilizes A egg, and you get an A child, and O sperm fertilizes O egg, and you get an O child. In this scenario, 25% of your children should be AB, 25% of your children should be A, 25% of your children should be B, and 25% of your children should be O. And look at the children here. 1 O, 1 A, 1 B. These numbers, 33%, 33 and a third percent, 33 and a third percent, 33 and a third percent. Those numbers would not be statistically different than this, right? Don't have any ABO siblings. don't have any AB siblings. My mom actually had a couple of miscarriages, so who knows? She could have had, my parents could have produced children who were AB, A, B, and O. Now, here's the thing. I am pretty sure, though I don't know if the data is out there to support this meaning, but I am pretty sure that almost everybody who's A is actually AO. So they're A, but they have an O hidden in their genotype. And most people who are B, they actually are BO, and they have the dominant B allele and the recessive O allele. I think this is how most A and B people are, just like my mom. The only scenario that really worked for mom and dad here was AO, BO. This is the most common scenario. So what that means is out there in the environment, there are lots of O's, but they're hidden away. You know, it's worth noting something. The most common blood type is O. The most common blood type in the US is O. It goes actually like this. O is the most common, then A, then B, then AB in the general population of the US. Now, in subpopulations, Native Americans, perhaps, they might not have that exact same relationship. But overall, O is dominant. Excuse me, sorry, I said that wrong. Overall, O is the most common. And you might think, well, wait a minute. How could O, which is the recessive allele, be the most common blood type? It's recessive. Well, remember, like I said before, just because it's recessive does not mean it is good or bad, common or rare. Those things are just not related. Remember that 6 fingers is dominant, but it's pretty rare. 5 fingers is recessive, but it is really common. The answer to this has to do with, it has to do with the population genetics of the human population, in which you realize that there are lots of O's out there circulating, right? No matter what, my children are going to have an O in the genotype. There's no doubt about it, because every sperm I make carries the O. That's all I have is O's. I can't give any A's or B's because I don't have A's or B's. If I had an A, well, then I wouldn't be O. I'd be AO, and my blood type would be A. So anyway, I wanted to just do this to defend my mom in case anybody is still wondering about the whole thing. All right, so very, very basic human genetics in a nutshell. Let's talk about some other things. There's a kind of genetic inheritance called pleiotropy, and this is where a single gene causes multiple physical effects. And it is sickle cell disease. If you inherit the sickle cell allele, it can cause sticky, fragile, abnormal shaped red blood cells. It can cause anemia, cause an enlarged spleen. I mean, there is all kinds of things that can happen that are the result of this. You have this one pair of alleles, and if you have the sickle cell allele, especially if that's all you have is the sickle cell allele, then that one gene, that one pair of alleles can have numerous effects in the body. So again, sometimes one gene influences many characteristics. Like I said, the whole sickle cell causes all of this stuff. It can cause weakness, anemia, heart failure, pain and fever, brain damage, rheumatism, kidney failure, pneumonia. Oh my gosh. I mean, here's a single gene that can have profound physical effects, and that's what pleiotropy is about. Now, there are also a whole bunch of genes that work together. These are called traits are called polygenic traits. Poly means many, and the genic means genes that work together to produce combinations of things. They work together. So things like eye color and skin color are this way. What they're showing down here in this graph here of the normal distribution of these things is they're showing there's 1, 2, 3 pairs of alleles that everybody gets 3 pairs of alleles. You can get both recessive, both recessive, both dominant, or more likely you're going to get some weird you're going to get some statistical combination of these. So this person here, yeah, sure, go ahead.
Okay, since we were talking about your beard earlier, I was waiting to see if you got here first. Is about, you know how you see the hair color for the top of your head is not the same dominant as the one in your beard. So you can have like a person has black hair and have like a fire red beard and everything else like that. How's that? Is that just left up to chance and like your beard facial hair color just shoots off an allele of red hair and doesn't have to, when nobody in your family has red hair, but your beard automatically says, you know, we're going to just make you have black hair and have a red beard, or vice versa, you have blonde hair and a dark colored beard?
Well, you know what? I don't really understand. I haven't spent enough time. It's been a while since I studied it, the inheritance of hair color. So I don't want to go out on a limb and say something that is wrong. So the bottom line is I think that the genes that produce hair, there's different kinds of hair in different parts of the body, like beard hair and pubic hair and head hair. These can be independent of each other to one degree or another, and so that sort of explains that there's I think different subset of genes that affect those different hair colors. But why you would have black hair and a blonde beard, which would probably not happen, but black hair and the different colored beard, I can't answer that. I don't want to. My little knowledge of it is likely to just get me into trouble. So now, again, I got to tell you, my beard, I put henna in my beard, so there's no reflection of the color over here is a reflection of the henna that I put in my beard for. Well, I put it in there telling myself that it's good for my hair, but the bottom line is it's just, it's something I know that people with beards do. They put henna in their beards, and I'm one of those people who does it. So it's not an accurate representation. Sorry, I can't answer the question. So anyway, but it's worth knowing there are polygenic traits in which many genes work together to produce a specific trait. So in polygenic inheritance, 2 or more genes combine their effects to produce a single phenotypic trait. So there are things like skin and eye color, the predisposition to alcoholism, predisposition to heart disease. They're not a single gene. Multiple genes and multiple alleles work together to produce a certain physical effect. But what about sex chromosomes? Sex chromosomes, the 23rd pair. Women have XX and men have XY. You knew about that. Some genes totally unrelated to sex determination are on the X chromosome. These are what they call sex-linked traits. So on the X chromosome, there are genes for things like color vision and blood clotting. These genes, when a man inherits an X chromosome from his mother, he inherits whatever alleles are present on her X chromosome. And unfortunately, his little stub Y chromosome does not contain those alleles. And so, look, if mom gives me, which mom did, some defective color vision alleles, I don't have another chromosome like I have in pair 1, 2, 3, 4, 5, 6, 7, 8, all the way up to 22. I've got 2 chromosomes. So if one has a defective allele, the other one probably has the non-defective form of the allele, and so everything works out fine. But in the XY pair, there are genes for important things like, as I said, like blood clotting and color vision that are on the X chromosome, but no complement on the Y chromosome. So a sex-linked trait is a trait that's related to your inheritance on the sex chromosomes. That's pair 23, right? So the X chromosome contains about 1,000 genes, and the Y chromosome has only 78 genes. Sorry, it's my cat that's doing that. Mouse? Yeah, you're making the thing go bad. You're making the PowerPoint move. So take a look, huge difference. The X chromosome, teeny tiny, 78 genes. Most of these are related to being a man and, you know, primary and secondary sexual characteristics and those kinds of things. So since men have only one X chromosome, they don't have a corresponding gene on the Y chromosome. So the gene on the X is expressed, even if it's recessive, even if it's defective. And again, I have to blame my mom for some of my color vision problems because I inherited a defective allele for some kinds of color vision on her X chromosome. Dad did not provide me with a chromosome that would help me out in this particular problem. So red-green color blindness and hemophilia are both examples of sex-linked traits. So look, I got to show you this picture. And I got to ask, okay, somebody in the chat line, do you see anything in here? I see nothing. I do not see any pattern of any kind in here. I don't see. Okay, 7, 7, 7. Well, you know what? Remember I told you I had defective color vision? Well, here's where it's showing up. I see nothing on this. I mean, if there's a 7 in here, I don't see it. Now, of course, I've looked at many of these charts, and when I look at many of these charts, I sometimes see things perfectly, but other times I see nothing. And on this particular one, I see nothing. And that's because I have certain kinds of color vision issues, such that when I look at this big, weird blob, I don't in any way see a 7 under any conditions. So I apologize. I don't know. If you knew that I had this color vision problem, maybe you never would have signed up for this class. So sex-linked inheritance means that some genes are inherited on the sex chromosome, mainly the X chromosome. So here's a female that has a normal clotting allele, capital H, and a defective clotting allele, lowercase h. But guess what? H is dominant over lowercase h, so she's going to have normal blood clotting. But now, take a look. Here's a man, XY. He got the abnormal or the, abnormal clotting allele. But does he have a normal clotting allele over here to counterbalance it? No. So he is going to get what mom gave him, no chance to cancel it out with what dad gave him. So he is going to inherit this allele, well, just one allele in this case, which is a different kind of thing. So the recessive allele on X and no matching one on Y. Sex-linked traits most commonly affect men. I mean, the mother is the one who gives us our X chromosomes. Remember, mom, all she has is X's, right? She's going to give you an X. In every egg, she's going to give you an X. Dad, half of his sperm carry the Y. I think we're done with this one. And I posted this one. There's lots of stuff here, lots of stuff, tons of material that I just went through that was just crazy. And I know that was tons and tons. So what is this, Travis, that's right here? A bunch of them?
No, those are glasses that have been developed to fix color blindness in people who are colorblind. I got a pair of them from my brother a few years ago.
Wow, interesting.
It's pretty amazing.
That is amazing. Have you ever heard of anybody who bought them? Oh, well, I'm not being buying them. They're cheapest one is 100, but still.
I got a pair of them from my brother who's green-red colorblind, and he claims it changed his life. He wears them every day. So really?
Well, you know what? That is really interesting. I appreciate you sending me that. I'm going to save that link. I mean, maybe for Christmas, I'll ask for a pair of glasses like that. Now, those glasses are not like vision correcting; they're just color correcting, right?
As far as I know, I think they're just color. I don't know if they've developed, because I got one from my brother like years ago. I don't know if they've developed prescription lenses for them too, but they have a specific test for your individual color blindness that you'll take, and then they'll give you lenses designed for your eyes, basically, to fix whatever your specific needs are. So
that is amazing.
I'm
I'd love to,
not trying to advertise or anything. I just thought they were cool. So.
Oh, that is cool. I'd love to try that just to see, because who knows? I mean, I might put them on and go, oh my gosh, there's a whole world of color I never saw. That wouldn't surprise me.
Yeah, like
yeah. So, Emily, I'm sorry that you're afraid of colorblind people. I don't know whether maybe when you were a young kitten, a colorblind person was mean to you. I don't know. You know, whenever I see people like, well, I guess it's mainly cats, right? When I see cats that are afraid of people, I think, well, it's because somebody was. So anyway, colorblind people, I wouldn't be afraid of them. I think that colorblind doesn't really affect much, I don't think, except it does affect how poorly I dress, right? Like sometimes I'll go out and my wife will tell me, oh my gosh, did you wear that out? Those two colors? I'm like, yeah, they seem to match to me. Oh my gosh, no. All right. All right, so look, I want to get, so that genetics PowerPoint is up there and available for you to study, study, study. This is the next PowerPoint I want to go to, and I want to take a break, though. I need to get up and stretch my legs, just like I'm sure you need to. I'm just trying to drag this over here a little bit. We are going to take a 10-minute break. Okay? 10-minute break starting now. It says 2:30 on my computer, so I'll be back at 2:40. I'm going to go walk around and see if I can regain any sensation whatsoever in my left leg. All right, be right back. All right, folks, I'm back. I'm going to get this PowerPoint booted up, but I'm happy to answer any questions people have about any of the material we've covered so far. Lots of material, tons of stuff.
I have a question for you.
Sure, go ahead.
So when I was taking biology, intro to biology 101, I think it was, we had to do the Punnett squares. And so the one I did on was about my wife and me. She has freckles, I don't. So preferably freckles will be dominant, right?
Freckles is dominant, right?
My daughter has not had freckles yet, and she's like 6. So
your wife has freckles, and your daughter. We would think if your wife has freckles, then no. Here's the thing. Sorry, Punnett Square should have told you this, right? Just think about this for a second. Your wife could be she has 2 alleles for freckles. It could be positive, or it could be positive, negative, right? She could have both of her alleles could be the freckle allele. Or, which is more likely, she has the freckle allele and the non-freckle allele. But because the freckle allele is dominant, she has freckles, right? Now, half of her eggs carry the freckle allele, and half of her eggs carry the non-freckle allele. Okay, so you are non-freckled, so all your sperm carry non-freckled alleles. So here's what it means. If one of your sperm fertilizes a freckle allele egg, then your child would have freckles. If one of your sperm fertilizes a non-freckle egg, then half of your children will have no freckles. So the Punnett square would tell you that that in your particular case, 50% of your children should have freckles, and 50% of your children should not have freckles. Even if your daughter had freckles, it wouldn't give us that much insight into what exactly is going on with your wife, because she's has freckles and the freckle allele is dominant, so that gives her freckles. But the fact that your daughter doesn't have freckles, it tells me that your wife is what they, again, the word is heterozygous. She has one allele for freckles and one allele for non-freckles. And so that means that 50% of your children should have freckles and 50% of your children won't have freckles. And that exactly, I don't know how many children you have. I know you have the one child, Astrid. And, but, if you decided to have a bunch of children, then maybe in the end, you'd get, I still think in the end, even if you had 100 children, which I wouldn't recommend, but if you had 100 children, you would 50% of them would have freckles and 50% of them would not have freckles. And that's just based on the observation that your daughter doesn't have freckles. So despite the fact that your wife has freckles, she must be heterozygous, right? You must be able to give your daughter the non-freckle allele. And the only other possibility, Kendall, is that that she's not actually the mother of your daughter. Think there's any chance of that? Could there have been a switch in the hospital or something bizarre like that? I'm only kidding, of course, but the bottom line is there's the story, and it doesn't make it makes total sense that your daughter wouldn't have freckles. There's a 50-50 chance that she would.
So
go ahead.
I have kind of a question along the same line. So so when you say it's 50-50, is it like exactly? Just because I've always been curious, because my mom, like I said the other day, I'm like one of 6 kids, but 4 of us my siblings have blue eyes, and then the other 3 we have like brown eyes. And I know blue is technically recessive, right?
I don't know that. Is it? Is blue recessive?
I thought it was. I'm pretty sure
this
brown
is
eyes are dominant. That's how I've always learned it.
this is a polygenic trait, right? So there's multiple genes involved in creating this color. However, let's just go back to what your original question was, which is it possible to have a number that's different than 50%, right? Because that's what you were asking is, look, you said it would have to be 50-50. Does it have to be exactly 50-50? And the answer is this. Statistically, mathematically, it's 50-50, right? Just like if you toss a coin 100 times, you expect to get 50 heads and 50 tails. That's what you'd expect to get. But if, let's just say you toss a coin 3 times, would you be astonished if 3 times in a row you got heads? I don't think so. I think you'd go, well, okay. That seems like, it's not what I would expect, but what would you expect? If you toss a coin 3 times, you could get 3 heads, 3 tails, 2 heads, 1 tail. I mean, there's different combinations. So to go back to the original answer is, yeah, statistically it's 50-50. But does that mean that you couldn't have a variation from 50-50 in a small sample size? Of course you could, right?
Okay.
Of course. My parents, the chances that they would have, remember, based on their genetics with the ABO blood type, right? The bottom line is that with the ABO blood type, my parents should have 1/4 AB, 1/4 B, 1/4 A, 1/4 O. But if they had all O children, that would be statistically possible, right? Even though they're supposed to have 1/4. If they had 100 children, which I hope they wouldn't, right? I could not keep track of people's birthdays and all that. They had that many children, well, then you would sort of, and they were all O's, you would go, oh, no, this is bizarre. But, it's a statistical thing. So Kendall and his wife may have all non-freckled kids if they have 5 kids, right? And that wouldn't really be a statistical deviation from the 50-50. It's a small sample, that's all, what it comes down to. So we did a little Punnett Square with the earlobe thing. Yeah, no, but that's a good question. And I mean, I don't know. I think that as far as Abril is saying, I'm really pretty sure that handsomeness and ugliness is a polygenic thing. A whole bunch of genes contribute to whether you are beautiful or beauty challenged. Let's put it that way to be nice, okay? So I'm not. So I guess what Abril, you're saying that you got the beauty gene and your siblings all got the polygenic combination that makes them less beautiful than you, and that doesn't surprise me one bit. Okay. All right, back to PowerPoints. What else? How do you teach in a virtual environment without PowerPoints? I mean, is there a way? I mean, I know other people are taking other virtual classes. I mean, when people are taking these other classes, what do they? I mean, how do they teach? I've not had the opportunity to really experience it very much to know what's going on. I think most classes like this do PowerPoints. I'm gonna honestly. I'm telling you, I'm not really a PowerPoint person, but I've had to become a PowerPoint person because I don't have a board to write on, and I don't have an overhead projector to use. In my normal classroom, I would have this technology that I could use, and I don't really have that technology at my fingertips now. I mean, I am hoping that sooner than later, we get a vaccine for COVID-19, and then we go back into a regular classroom, and hopefully I remember how to teach in a regular classroom. And then I also hope, just between you and me, that we don't forget COVID-19 and we start investing money in getting ready for the next big virus that's coming out of the rainforest at us. We don't disband the entire, once we get COVID-19 conquered by vaccine. I hope we don't just say, okay, that's it. We're never going to disband all this. No, we need to realize that these kinds of pandemic viruses are possible in the future, and we need to prepare for them now. Anyway, that's just my opinion. I'm on my soapbox. So this was called Very Basic Embryology because that's what it is. We're going to boom through it. Let's talk about sort of stages of development. Way back when we talked about development. Remember that in our particular case, we start out with gametes being formed, sperm and eggs. Under the right consensual romantic circumstances, fertilization occurs. Then the embryo forms. It forms a zygote. It undergoes cleavage to make more cells. It forms a gastrula. It forms organs, and then it starts to form, well, it forms tissues and organs. I mean, really, I should go back and correct this and go gastrulation. Move this over here. Tissue specialization, organ formation, and growth. That is more accurate than what I had before. Sorry. All right. We're going to look at all of these today briefly. So you know what it's like to the process of meiosis that produces a sperm that looks like this. It has, of course, this is a human, so it has 23 DNA molecules in it. It has a head that's got, you know, a nucleus and cell membrane and all that, a little packet of enzymes up here, a neck or mid-piece or neck here attached to a mid-piece that has a lot of mitochondria. Those mitochondria are all about driving this tail, and you remember very well, I'm sure, how this tail is built, at least in a very basic way, because it's built with microtubules in the same way that cilia is built, but it's much longer. And the goal of a sperm, obviously, is to work its way through a serious gauntlet to fertilize an egg. So what does that really look like? We don't need to go into great detail, but of course, this all starts in tubules inside the testis. There are sperm mother cells here, spermatogonia. They will, when they are exposed to the right hormones, starting at puberty for men, because men don't make a single sperm until puberty, not like women that set aside their eggs when they are in their mother's womb. Men make fresh sperm every day, hundreds of millions of them. So they start with a diploid mother cell that becomes a diploid primary spermatocyte. That divides. This is meiosis 1, okay? And then we end up with haploid cells, and each of those divides again. This is the reduction division where we go from diploid to haploid. And here, all we're doing is that it's not, what's actually going on with this N is there's 23 replicated chromosomes here. But we don't want replicated chromosomes. We want individual chromosomes. So those come apart in meiosis 2, and now you have 4 sperm cells and 4 potential sperm and 4 sperm cells, and every one of them is haploid. Right? So basic development of sperm. Remember, in meiosis, we start out with a single diploid cell, and we end up with 4 haploid cells in most circumstances. It's not true for eggs. Again, here's just some weird and bizarre drawings of sperm that have been out there. I don't know. Some of these, I like this one because it looks like it's wearing glasses. And oh, I guess this one sort of has a beard. But anyway, bottom line is that I've seen sperm under the microscope, and none of them seem to have eyes or mouths or any of that kind of stuff. What about the development of an egg that can be fertilized? Well, in the case of meiosis for eggs, only one ovum capable of being fertilized is produced. There are 3, maybe 2 polar bodies that contain very little cytoplasm and eventually degenerate. So in this case, this is an exception. Rather than making 4 gametes, when eggs are developed, they absolutely want to produce one big plump gamete that has most of the cytoplasm. And then they produce 2 or 3. I mean, 3 is the number you expect, right? Because you're supposed to produce 4. Produce 2 or 3 of these things called polar bodies that could not develop into an embryo. They have only a nucleus and a tiny bit of cytoplasm, and they just get flushed out of the body. So again, we start out with a diploid mother cell in the embryo, and then remember that diploid mother cell starts meiosis. It puts all of the chromosomes in synapsis in prophase 1 of meiosis. So it's basically stuck here for a period of time, arrested in prophase of meiosis 1, right? Then, after it's been, as it's going through its development, it starts to move into producing actual haploid cells. Here's one, the big egg that's going to be the big fertilizable egg, and here's the first polar body, dead, this will die. Then it goes through that, the entry of sperm triggers the completion of it, and now what you have is one big plump cell with lots of cytoplasm and all the organelles you need to be a complete cell, and a second polar body. And you know what? Sometimes they say that this polar body divides, but I don't really think that happens most of the time. So now you've got the egg that has been fertilized. It has sperm nucleus in it, and it has egg nucleus in it, and that's going to become the embryo. But notice what happened is, instead of this being divided into 4 equal sized things like it is in sperm, one of the cells gets to keep most of the cytoplasm and become a big plump egg cell. Okay, so here it is. This is called unequal cytokinesis. So you remember what cytokinesis is, okay? It is where the cells divide, and I usually say divide in half, but that's not what's happening here. Unequal cytokinesis produces, what have I done here? Didn't mean to do that. Produces one big plump egg cell with most of the cytoplasm and 2 or 3 polar bodies that will die. They can't be fertilized or anything like that. So that word is unequal cytokinesis. And that's what happens here. Oh my God, look, I don't want that to happen. I want to get rid of that, I wonder. So this becomes part of the recording. Okay, so how do I get away from that? Oh, I need to figure this out. This is bugging me. No, that's not it. Turn off captions. I'm glad I figured that out. It didn't take hours. I don't think the captions would be that useful. Maybe they would be. But okay. We're back to work, right? Unequal cytokinesis. All right, so, again, human females set aside all the possible eggs they're ever going to use in their fetal ovaries when they're inside their mother's womb, and they sit there in suspended animation for many years, possibly. I mean, I think, I don't know, we could Google it, find what the average age of the beginning of menstruation is. It's probably like 11, and we say that those eggs, that the egg that you produced when you're 11 years old as part of your first ovulatory cycle is 11 years old. And obviously, the older you get, the longer a cell that's going to become an egg has been sitting in suspended animation, and it kills lots of eggs. I mean, you set aside 5 million eggs, but by the time you're actually at puberty, I mean, you maybe have 500,000 eggs. And remember that all you really need is probably about 650 eggs, unless you are planning to be in the Guinness Book of World Records. And then it would be good if you had maybe more than that, so you could ovulate lots of eggs and get those fertilized, so you can get just that magic number 70 and be in the Guinness Book of World Records. So oogenesis is the formation of the egg, and again, we end up with one big plump egg. Spermatogenesis, the production of sperm, we end up with 4 sperm. 1 oocyte, 4 sperm. The progenitor in eggs divides once. The progenitor cell, the mother cell for sperm, divides thousands of times. There's resting periods, like in the case of the formation of eggs. There's times when things are sort of stopped and they are waiting to be fertilized versus continuous. Men are continuously producing sperm. And in oogenesis, the actual completion of meiosis happens after fertilization versus before fertilization. Men better get their sperm lined up and ready to be fully functional before fertilization. But you know, I gotta say it. It is amazing, the story of fertilization. It absolutely is. Years ago, I worked for a company and wrote textbooks and other kinds of educational materials for them, and I got a call one day, and they said, "Steve, we want you to write a sort of just one-page, quick, oh wow kind of thing about something related to the male reproductive tract." And I had written, for these textbooks, I had written a sort of page at the beginning of a chapter where it was designed to sort of get people like excited or interested about the material in that chapter. And I'd done it before, and people liked it, so they called me and they said, "Look, would you do this for this book that's being written on male reproduction?" I said, "Sure." And I said, "What do you want me to write it about?" And they said, "Oh, we don't want to tell you. You tell us, because the ones you produced so far are dynamite." Okay, so I said, "All right, I'm going to write about the lucky sperm." And I said this off the top of my head, and it kind of got a weird look from the editor and the assistant editor. And I said, "Don't worry, it'll be interesting." So here's what I found out when I started to do research. I found out, number one, that sperm cannot swim in the male's body. They cannot swim. They are paralyzed inside of the male's body for good reason. Because you have millions of sperm packed together. And those millions of sperm that are packed together, if they were all whipping their tails, it would damage other sperm. So they're paralyzed inside the man's body. They actually don't start swimming until they leave the man's body, and under the appropriate, totally consensual romantic circumstances, they find themselves in the vagina. Then they can swim. But inside the male's body, no, they're not allowed to swim because they will damage each other. And when the man ejaculates, he's ejaculating about 500 million sperm. Most of those sperm are doomed. They will die. Because, among other things, the vagina is not necessarily a really sperm-friendly place. It's not a friendly place for most microscopic things. It's pretty acidic. That kills a lot of bacteria and damages viruses and kills funguses and actually kills sperm. So lots of sperm get killed right away in the vagina. Some that are left start swimming up through the cervix, up to the uterus, heading for the fallopian tube, They have to get in the fallopian tube and all the way up almost to the end of the fallopian tube, like 2/3 of the way up, let's say, towards the ovary before they're likely to encounter an egg, right, under the right circumstances. So they start swimming. Now, this is a gauntlet. There's white blood cells, there's mucus, there's all kinds of things that can prevent sperm from getting anywhere near an egg. It's possible that when a woman is ovulating and her body would really try to set the stage for fertilization, that she might diminish that stuff a little bit. But overall, it's a gauntlet. Now, when some lucky sperm gets in the oviduct is a tube, and that tube is, that oviduct tube has cilia in it. And the cilia are beating in this direction, can move an egg down to towards the uterus. The sperm are swimming this way. They have to swim upstream against a current being produced by cilia. Now, I forgot to tell you this. I researched it, went to a cadaver, measured it, and found out that the distance that a sperm needs to swim from when it enters the vagina to where it fertilizes the egg, that distance is the equivalent of swimming 5 miles. But remember, sperm don't get to practice swimming. They don't get to practice. They're in the vagina. Now they can swim. Now they need to swim the equivalent of 5 miles up the oviduct against cilia beating in this direction to move the ovum down. And so, remember I said that in that ejaculation, there's highly 500 million sperm, a huge number. When they have counted sperm near the egg, there is less than 500. That means that 1,000,000,000,001% of the sperm get anywhere near the egg. I mean, it is a huge gauntlet. And really, the sperm that eventually gets there is either incredibly lucky that it got anywhere near an egg, or it's like a ninja sperm that has somehow managed to get through all of that. It's like that Dwayne "The Rock" Johnson guy jumping from one skyscraper to another or something like that. So when a sperm finally does fertilize an egg, it's 1 in 500 million, and most of those 500 million are lost or dead or confused. I don't know. So the question is, how do sperm know where to go? And really, there's multiple hypotheses about these. There's a hypothesis that suggests that when a woman is ovulating, there are pheromones released from her ovaries that are there in minute amounts that sperm can sometimes track and find. That's one hypothesis. Another hypothesis is that it's purely random. Sperm go all over the place in the female reproductive tract. They go up the wrong fallopian tube. They go, and that's again why maybe so few ever get anywhere near an egg. So anyway, I did a little research, looked it up, was kind of amazed at how complicated it was. And I think it's me who first coined the phrase, I'm trademarking it, who first coined the phrase ninja sperm for the sperm that is lucky enough to get anywhere near the egg. Okay? Now, in this picture, I see an egg and I see all kinds of sperm all over it, and that doesn't necessarily support my story. But so the sperm and egg unite to form a diploid zygote. That's what it's called. The zygote is like it's like a fertilized egg that's ready to divide. 23 chromosomes of the egg unite with their homologous mates in the sperm. Remember, the sperm brings one member of pair 1, one member of pair 2, one member of pair 3, one member of pair, all of these pairs until 23. The egg has one member of pair 1, one member of pair 2, all the way up to one member of pair 23. 23 plus 23 equals 46. 46 is the normal DNA number for a human. Somebody going to ask a question? I hear a microphone. Nope. All right, that's fine. So this is an image of the sperm sort of digesting its way through the outer covering of the eggs. Eventually, it releases a sperm nucleus. There's an egg nucleus. The two unite to form the nucleus of the zygote. Once that zygote nucleus is formed, then the egg is ready to divide. And it kind of looks like this. You start out with the zygote, divides to form 2 cells, divides to form 4 cells, 4 cells form 8 cells, 8 cells form 16, 32 morula, then the blastula, then the gastrula. In the gastrula, we see these things, the primary germ layers, which I want to talk about, of course. So, what's actually interesting is that generally, as you can see, here's where fertilization happens, near the ovary. Then, once fertilization happens, thanks to the cilia that line the oviduct, they start to go, the fertilized egg starts to be moved down. It's already dividing and dividing until it gets, and it implants in the uterus. I think the first divisions in humans start in about 5 hours after fertilization. So it starts dividing right then and there. Now, when it's dividing, this early time, the cells are generic cells, and they're producing generic cells. It's a little bit further down that we start to see cells beginning to specialize. Eventually, there are 3 cell layers that form. And these 3 layers that form are the endoderm, mesoderm, ectoderm. Now, ignore goldenrod packet 34.5, sorry. But these 3 cell layers form when we start to get this gastrula formation. And gaster means that actually what's going to start to happen here is a tube is going to form that's going to form the gastrointestinal tract. And that's going to result inside the cell with these 3 layers of cells. Now, take a look. The innermost layer that's going to be associated with the digestive tract is called the endoderm. It forms the lining of the digestive and respiratory tracts. It forms the tonsils, liver, pancreas. It forms parts of many glands. Okay? Endoderm. Mesoderm. Oh, let's go with endoderm. Ectoderm is the outer edge. And it, you're going to not be surprised, forms the epidermis of the skin derivatives like hair, nails, etc. It forms the tooth enamel. It forms the entire nervous system. And it forms portions of most sense organs, which of course are related to the nervous system. The nervous system forms entirely from ectoderm. In between the inner layer of cells that form the lining of the gut and the outer layers of cells that form the outside of this embryo, there's a layer called the meso or middle layer. And the mesoderm forms all connective tissues like cartilage, blood, and bone, all connective tissues. Mesoderm forms all muscle tissues. So there's 3 types of muscle tissue. Remember, there's smooth muscle, cardiac muscle, and skeletal muscle. They're all formed from mesoderm. And then mesoderm forms parts of all these internal organs. I mean, in some cases, these organs have some of these organs are going to have pieces of endoderm associated with them too. But I always think that when I think about endoderm and I think about the digestive tract and the respiratory tract and a lot about the kind of tissue called epithelium, when I think of ectoderm, I think of the epidermis of the skin, which the skin is the largest organ of the body, covers the entire body it's supposed to. And I think about the nervous system all coming from ectoderm. And then when I think about mesoderm, I think, wow, all connective tissues. I mean, connective tissues are one of the most common tissues in the body, and they make up bone, blood, cartilage, ligaments, tendons, blood, lots of connective tissues are huge. We won't get to connective tissues today. We'll get to connective tissues on Monday. You're not going to want to miss that. All muscle tissue, amazingly enough, we might not impossibly get to muscle tissue today. All right, so these are what are called the primary germ layers. Okay, and honestly, I want to put that these are usually called primary germ layers. Now, germ in this case means to germinate or form. And these 3 primary germ layers will develop into all the parts of the adult embryo. Primary germ layers is what they're called. All right. So I'm tweaking this, so I will of course repost it. Right? So here's a document that I often used when I was teaching very basic embryology in the classroom. And I just want to mention it. Look, up here in the top, male gamete, female gamete. These were produced, of course, by meiosis. Now, should they come together under the right circumstances, they form a sort of N plus N embryo, right? Where you've got the sperm nucleus and the egg nucleus. Those will fuse together to form the zygote so that you have a complete basic nucleus, and now it's ready to divide. It'll divide to form 2 cells, 4 cells, 8 cells, 16 cells, 32 cells. The first kind of named embryonic stage here is called the morula. And the morula is a cluster of approximately 16 to 32 cells. And I don't know, I've always wanted to do this. And so I'm going to do it. It's called morula after the Latin word for mulberry, and really, when I look at mulberries, they remind me exactly of like blackberries. They look like a cluster of little round balls stuck together, and that's what this particular embryological stage can look like. It can look like, there's no reason to go so far down. I think I could go right here and take a look at any of these images here, and I could see why they might consider it that it looks like a mulberry, right? All right, sorry, had to do it. Got my internet fired up right in front of me, and so when we're talking about this right here, I wonder if this could work. Hold on, gotta try it. Sure that this is gonna make a big mistake, but I'm gonna totally screw things up here. Maybe not. Yeah, I did totally screw things up. All gone. So I did screw things up. Well, I'll fix it before I figure out how to put it together. But now, unfortunately, I don't have access to this document. So give me a second. Sorry, folks. That was crazy. I should definitely not have done that. It just took me a second. Reclaim that document, I think. Hmm. Oh, come on. Technical difficulties created by the instructor. And there was no blame this technical difficulty on anybody but me. Now, thank gosh, that mulberry's gone, so I can't possibly decide to paste it in again. So, okay, they call this a more because it looks like a mulberry. Now, what's going to happen is this is a big dead cells, but cells are going to start moving away from the center, and they're going to form a hollow ball stage called the blastula. So it goes, like it goes zygote, 2 cell, 4 cell, 8, 16, 32 equals morula. Morula develops into blastula. Blastula has outer cells like this, and then that will start to fold in here and form the gastrula. And of course, like I said, the gastrula is referred to the primitive gut. Okay, that's because this little puckering and folding here, the primary blastopore, it's going to eventually poke all the way through the embryo, and now it's going to form 3 layers of cell: the endoderm, the mesoderm, the ectoderm. Ectoderm, mesoderm, endoderm. Endoderm, endoderm, mesoderm, ectoderm. Now, give me a second. I don't want to write anybody a letter right now. Just some quick images while we're talking about this. Well, unfertilized egg, fertilized egg, 2 cell, 4 cell, 8 cell, morula, blastula, gastrula. But let's just take a look at some actual images. Now, this is starfish development. Here's an unfertilized starfish egg. Here is a fertilized starfish egg. Here is an 8 cell stage. Here's a blastula, 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 but let's look at some individual ones here. This one is a very big gastrula. Remember I said that there's a point where it starts to fold in like this, and actually this is going to fold in all the way through here. So before this puckering and folding happens, it's called the blastula. So let's just, for fun, here's a blastula. Cells on the outside, hollow inside. Oh, that's a large, two-cell stage. Oh, I can sort of see a little dot in there. It must be the nucleus. Another gastrula where it has folded even further through. Actually, this is going to bend up here, and it's going to poke through over here, and it's going to form one end of the digestive tract. Here's another blastula, hollow ball stage. Another even more advanced gastrula. Another blastula. Well, anyway, if you want to see these embryological stages, there's tons of really cool stuff posted out there. Now, where I was heading with this mostly was down to the point where we go from, fertilized egg and zygote, 2-cell, 4, 8, 16, 32, morula, blastula, gastrula, and eventually this tube forms all the way through like this. And now we have the first layers of cells in this embryo. And those layers of cells, of course, are going to form the primary germ layers. Now, we've already talked about this, so we don't need to go there. Again, we were talking about development in humans is a little harder to picture and find. But sea urchins and starfishes and things we're actually more closely related to those than you think, and they have a very similar developmental pattern, right? And eventually we get there. All right, now, all of this, an awful lot of what I was doing here today was about this. The reason is because this is where cells start to specialize in the body. Cells start to specialize in the body, that's when we start talking about tissues. Oh my gosh, one of my favorite topics, tissues. So notice that here we're actually mentioning tissues like epithelial tissues and muscular tissue and connective tissue and nervous tissue. So that's where I was headed with this very, 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 very basic embryology, I have to say. Okay? I was heading all the way to there. Now, last slide in here is designed to introduce tissues. So here's a document that I'm providing for you in this PowerPoint. It's also in the next PowerPoint, and it is histology, a scientific study of tissues. Tissues are one of the 4 categories of specialized cell types in the body. 4 categories of specialized cells in the body. Here's the amazing thing. There's probably, and honestly, I want to look this up and find somebody I trust and find out what the exact numbers are, but I think that there are approximately 150 different types of specialized cells in the body. 150 or so. I could be high, I could be low, but I think that that's about right. But what histologists have done is they've taken all that 150 cells and they have categorized them into 4 groups. And here are the 4 groups: muscular, epithelial, nervous, and connective. Now, I got to tell you, I use this acronym M E pretty bad E, huh? I use this acronym MENC. I'm getting better at writing with this thing, but not much better. Muscular, epithelial, nervous, connective. Muscular, epithelial, nervous, connective. The 4 categories of specialized cells in the body. MENC. Muscular, epithelial, nervous, and connective. Now, let's just look at the definition of a tissue for a second, okay? Tissue is one of the 4 categories of specialized cell types in the body. A tissue can be a region, a layer, a cluster of cells with similar shape and function. And tissues are usually derived from a small number of embryonic mother cells. And what I really mean here is that, there could be one cell in the embryo that forms the entire heart, or entire big time portions of the heart, a single cell. So when we think about tissues, the most common kind of definition of a tissue is something like this. It's a region, a layer, a cluster of cells with similar shape and function, but I think it's essential to say, look, it's one of the 4 categories of specialized cell types in the body. I mean, after all, I said there were 150 different kinds of cells. 150 different kinds of cells. They can be divided into 4 categories of cells: muscular, epithelial, nervous, and connective. Now, take a look at their origins, because this is what we were just talking about: primary germ layers, etc. All muscle tissue comes from mesoderm. Epithelial tissues, notice, can come from all 3 primary germ layers. Nervous tissue, I'm sure you recall, all comes from ectoderm. Connective tissue all comes from mesoderm. So we've got embryological origins. Now, this chart, super simple, basic. We're going to go into great detail. Muscular tissues, here's some important functions: movement, support, protection, contraction. And the notes over here, skeletal smooth muscle, well, that's because it turns out that there's actually 3 types of muscle tissue. Skeletal muscle, that is involved in moving the skeleton around. Smooth muscle, that's involved in creating motion in organs. And cardiac muscle, which you can probably guess only exists in the heart and is there to make heart contractions happen. So these notes are a little bit random, but oh well. Now, epithelial tissues, as I said, came from all of the 3 germ layers. And when I think of epithelial tissues, I think, wow, you can never go wrong by saying protection, because epithelial tissues are very involved in protection. But notice they're also involved in secretion and absorption. Epithelial tissues cover things and line them. Like when you eat food, that food is in constant contact with epithelial tissue as it moves from your mouth into your esophagus, into your stomach, into your small intestine, large intestine. It is the cells that it is in contact with are epithelial cells. And so my notes, again, kind of random. When I think of epithelium and I think of the largest organ in the body, I think of the epidermis, which is native epithelium. And then endothelium is a kind of epithelium that lines blood vessels and the heart, etc. But we'll talk about other kinds of epithelium for sure. Nervous tissue all comes from ectoderm. You knew that. And functions, well, I got to say it, that you probably don't remember because it was so long ago that we talked about this SAR. But SAR stands for sense, analyze, respond. Sense, analyze, respond. And we're going to talk about how the nervous system is divided in a way that it's pretty clear that there are portions that are sensory for sensing things. There's portions that are motor for moving things. And then there's portions that are central for analysis. So sense, analyze, respond. But when I think of the nervous system, I think of communication, thought, protection. In the nervous system, there's 2 main types of cells. Oh, there's a bunch of specialized cells, but there's 2 main types. The kind that are called neurons, and neurons do this. They carry a nerve impulse from point A to point B. That's what that means. Neurons are like wires that carry a specific nerve impulse, a specific bit of nervous information from one place to another. And then there are the support system cells of the nervous system that just do a ton of different things. Super important. I mean, really, just as important as neurons. And those are called neuroglial cells. All right. The C stands for connective. All connective tissue comes from mesoderm. Well, when I think of connection, of course, when I think of connective tissue, I think of connective, like connection, like the way that bones connect to muscles, the way that there are fibers throughout the body that hold and connect things together. Then support, protection, immunity. Well, remember, blood is a kind of connective tissue, and that means white blood cells, etc. Support and protection. Well, the skeleton, bones are a support tissue, and they support the entire body, give it its shape, etc. And then take a look at there's a huge amount of diversity in this group, a huge amount of diversity, because in the connective tissues, we see bones, blood, ligaments, tendons, fat tissue, cartilage, the dermis of the skin, etc. Lots of different kinds. Now, down here at the bottom is a chart that talks about some of the really basic characteristics and the comparisons between these. And so it's worth taking a look at now. When we look at epithelial tissues, we notice that the cells are tightly packed together. When we look at nervous tissue, tightly packed together. When we look at muscle tissue, the cells are tightly packed together. But connective tissue is a little different. It's really common in connective tissue for there to be cells that are, forget it, Steve, to be cells that are scattered around. I will show you some images, right? So connective tissue has cells that are scattered around. They're not tightly packed together. How can they be scattered around? Well, they're actually scattered around in a non-living protein matrix. And that non-living protein matrix, it's called the matrix. It's non-living protein stuff. And it is what the cells of connective tissue are scattered around in. You know, one matrix you may have heard of is plasma. Blood plasma is a liquid matrix. And the cells in blood, red blood cells, white blood cells, those things, white blood cells, platelets, they float around in that connective tissue matrix liquid. But in bone, the matrix is hard and mineralized, and the cells are scattered around in it. You're going to see examples of this when we talk about connective tissues. So there is a connection there, no pun intended. So notice if you are tightly packed, you can't have any matrix. No. Only if you are loosely and scattered do you have a matrix. How about this one, vascularized? This means blood vessels. Are there blood vessels in this tissue? Well, notice in nervous tissue, yep. In connective tissue, yep. And in muscular tissue, you bet, blood vessels. But what about epithelial tissues? No. And I'll tell you why. Epithelial tissues, as we're soon going to learn, are fragile tissues. They are constantly being replaced and remade because they are constantly being lost. Epithelial tissues form the epidermis of the skin, remember? And the skin's always coming off. Epithelial cells form the lining of hair glands, where hairs are formed. When hair falls out, epithelial cells fall out. So it wouldn't be a great place to put a blood vessel since it's kind of unstable. That doesn't mean that epithelial tissues don't need blood. They do. But most epithelial tissues are tightly connected to a connective tissue, and you're going to find blood vessels there. I'll show you some images. So the one we talked here, other one we didn't talk about, multinucleate. Well, notice that multinucleate means more than one nucleus per cell. In epithelium, no, generally no. Nervous, no. Connective, no. In one kind of muscle tissue, though, skeletal muscle, the kind that forms muscles like the biceps brachii, those can be multinucleate. In fact, a skeletal muscle cell in the biceps brachii could have 100 nuclei, no problem. But that's a little bit unusual. Lastly is this one, free surface. And what free surface sort of means, is Is it possible that you could actually touch this tissue with your finger? Is it going to be exposed to a Twinkie if you eat it? Is it possible that you may actually be able to see it? And the answer is in epithelium, yes. Because when I look at you, I see epithelium. I see the keratinized, stratified, squamous epithelium of your epidermis of the skin. But am I likely to be able to directly touch nervous tissue, connective tissue, or muscular tissue? No. In fact, look, it's never good. If something happens to you, it is never good if you see nervous tissue, connective tissue, or muscular tissue. I mean, look, I see connective tissue all the time because I bleed all the time. I don't know why. It's because I play with cats. And like my mom used to say, if you play with cats, you will get scratched. So I have connective tissue leaking out all the time. And yet, I sometimes don't even know it. Somebody will come up to me and go, "Excuse me, sir, you're bleeding." And I think, "Oh, wow, thanks for telling me." And I realize you're really not supposed to see connective tissue. You don't generally see adipose tissue or blood or bone or tendons or ligaments. No, you're not supposed to see that. There's no free surface. It isn't sticking up into an empty space. No, we're not supposed to see nervous tissue. We're not supposed to see connective tissue. We're not supposed to see muscular tissue. Oh, we'll see them because we're studying these tissues. But in a generally daily basis, if you look down at your hand and you see muscular tissue, that's bad. You shouldn't be seeing muscles. I remember when I was a kid, I got a really deep cut right through my finger. It went all the way to the bone. The knife dragged off the bone. And I looked into that crack and I could see bone and ligament, maybe tendons, maybe muscle, etc. And I knew that was bad, that it was time to go to the hospital and get all that stitched up. Anyway, we'll continue to talk about some of this stuff, but this particular one is done. I made a couple of small changes to this, like I screwed up the whole mulberry thing. But I hadn't made very big changes, so I'm actually probably not going to repost this one, okay? Now, where am I? What's going on here? Where am I? Yeah, that's me. Okay, so the next PowerPoint that we're going to look at after we take a break, because I need to get up and move a little bit, okay, this is the next one that we're going to look at. We're going to start talking about epithelial tissues. All right, but I got to get up. I can't even feel my left leg again, okay? So on a 10-minute break. Oh, textbook said 200 specialized cells. You know, I believe it. I'm not totally, and I think 150 may be conservative. All right, 10-minute break. I'll leave you with this smooth jazz. Hi, Emily. All right, I'm back. Whoa. Well, we've accomplished a lot today, at least on paper we have. I hope that people are going to realize that really need to go back and take a look at these PowerPoints again and study them. And I'm not saying memorize them, but don't really need to memorize anything in this class, do you? Because everything is open note, open book. Which isn't an ideal class, but hey, it is what it is, right? Okay, tissues. Let's talk about tissues. We're going to start with a little introduction to tissues, and then we're going to start talking about one of my favorite tissues. Oh, I don't know if it's my favorite of the four, but it's certainly one of my favorite epithelial tissues. So, you and I have talked about this before. The correlation between structure and function is one of biology's most fundamental concepts. Form follows function. We've done that again and again. Animal structure, like has a hierarchy. We've talked about this at great length before. We started with atoms. We went all the way to the biosphere. Organelles, cells, tissues, organ systems. This is mostly the sort of stuff of Bio 156. But, when we talk about, say, tissues, we're going to talk about, okay, here's a specific cell, a specialized cell, a muscle cell. That muscle cell is part of a layer of muscle tissue. That muscle tissue is a layer of cardiac muscle tissue in the heart. And of course, the heart is part of the cardiovascular or circulatory system. And the circulatory system is one of the major systems in the body. The organism, of course, is many organ systems working and functioning together. There's no real news there. We're just introductory stuff. So tissues. Histology is the study of tissues of the body. Tissues are groups of cells that work together to form a specific function. MENC, remember what MENC stands for, muscular, epithelial, nervous, and connective. And organs are formed from tissues. In fact, I usually think that a typical organ has all four tissue types working together. Like in the heart, there's cardiac muscle tissue. There's epithelial tissue that lines the heart and forms, the lining of the heart. There's nervous tissue that is connected to the heart so that we can sense pain in the heart and we can control heart function. And is there connective tissue in the heart? Well, in one sense, there certainly is, because in a typical heart, there's blood, and blood is a connective tissue. But more than that, there's tissues in the heart that bind the other organs together, and those are, of course, connective tissues. Well, here's a cross-section of the small intestine. It shows connective tissues, muscle tissues. There's epithelial tissue here. I see blood vessels here, and all blood vessels are lined, if not entirely formed, by epithelial tissue. Then there's, as I said, 11 organ systems that work together to perform life's function. Each organ system can be sort of dissected down into the organs that make it up. Like in the heart, in the cardiovascular system, there's the heart, there's arteries, there's veins, there's divisions of veins called venules, there's divisions of arteries called arterioles, and between the two, there's capillaries that are the tiniest microscopic blood vessels of the body, and we've talked a little bit about those before. Oh, here's some important terms in the study of tissue. Secretion, you know what secretion is. Secretion is the production and release of a fluid from a cell, but let's do this correctly. Secretions are valuable substances that are produced by cells. They have a function in the body, like mucus is a protective secretion. Like salivary amylase is a digestive substance. And I noticed that there was a tiny bit of confusion about the secretion thing. I mean, I think it's good to remember that there are many cells of the body whose job it is to make substances, valuable substances, and to release them to do some valuable function in the body, like saliva or mucus or digestive enzymes or hormones. Oh, we studied secretions. Absorption is the intake or uptake of fluids, ions, nutrients by cells. Intercellular, of course, means between cells. Extracellular means outside of cells. Intracellular means within cells. Matrix, this is something I mentioned a few minutes ago, is the intercellular material between adjacent cells, an important part of all connective tissues. Okay. We've already looked at this chart, so we don't need to look at this chart again. We are going to talk about epithelial tissue. Epithelial tissue covers the body and lines organs and cavities. Often epithelial tissues are sheets of closely packed cells that cover surfaces, line cavities, and tubes of internal organs. Look, when you look at another human being, what kind of tissue do you see? Epithelial tissue, because the epidermis of the skin is made of epithelium. General functions of epithelial tissue: absorption, diffusion, secretion, lubrication. So its goblet cells are part of epithelial tissue, and goblet cells, of course, make mucus. Under the right sort of circumstances, that mucus is a lubricant. Epithelial tissues are for protection. Now, general features of epithelium. Epithelial cells are closely packed cells, usually forming continuous sheets and lining. There's no matrix in epithelium. There's very little stuff outside the cell. The cells are tightly anchored together by specialized, tight and anchoring protein junctions. Epithelium is avascular. You don't want to put blood vessels in epithelium because it is constantly changing. I mean, look, the epidermis of our skin, remember, the stratum germinativum divides twice a day to make new epithelium. Epithelium tends to divide a lot because it needs to be replaced. Those cells that line our mouth, when we brush our teeth, we scrape off millions of those cells and spit them in the sink. And so it's required that that the mother cells do a lot of cell division to continuously replace those cells. Generally, epithelial cells have one free surface. And what that means is they have one edge that faces into an empty space, and that empty space is called a lumen. We'll talk about that in a minute. And then they have a base or basal surface that sits on a sort of glue that glues epithelial tissue to connective tissue called the basement membrane. So here's the thing. When we look at the epidermis of the skin, we know that we can touch it, and so there's some cells there that are on the surface. But then there's many, many layers of cells. Those cells don't have a free surface. They're attached to another epithelial cell. And at the very bottom of that, there's the stratum germinativum, which is glued to connective tissue underneath it by the basement membrane. So here's kind of a typical epithelial cell. Here's the cells themselves. Okay, now 1, 2, 3, 4, nucleus, 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 cell membrane, etc. Notice that in these cells, one end is free. This thing out here, where the cells stick into, the actual name for that is it's usually referred to, horrible, as the lumen, L-U-M-E-N. I'm not even going to try. This is the lumen out here, okay? And so this is like a free surface, meaning these cells are actually sticking leaking into now this could be the small intestine, and this could be food out here. This could be your mouth, and this could be food or toothpaste or who knows what out here, some potato chips. But at the other end of the cell, it is glued to connective tissue, and this double layer of stuff here is called the basement membrane. Some of this is secreted by the epithelial cell, some of this is secreted by the connective tissue cell, but it forms a layer of glue, really, that's called the basement membrane. Now, here's connective tissue down here, and this is the blood vessel. So if you're wondering, how can these cells get the benefits of blood, which means oxygen, taking away carbon dioxide, nutrients? Well, that's because this is a blood vessel that's very close, and nutrients will move out of it, and oxygen will diffuse out of it, and carbon dioxide will diffuse into it. And so there's always a blood supply nearby. That thin layer underneath the epithelial cells is the basement membrane. Now, here's a cross-section through a picture of the kidney, right? Or through an actual kidney sample, cross-section. What you're seeing here, this is the edge of a tube. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Did I get it right? Oh, actually, here's a tube too. You see this tube? This is a blood vessel. And this blood vessel is very thin. It's made of what's called simple squamous epithelium, which is just really one cell. It's thick. It's very thin, flat cell that wraps around and forms the blood vessel. The interior of a tube like this is called the lumen. Now, I didn't write this, and so whoever wrote it actually has better writing than me. So the lumen is the empty space inside of a tube. In the kidney, the lumen here is going to be filled with a very dilute urine that is filtered out by specialized filtering structures in the kidney. But this lumen is the empty space. And in order to understand epithelium, you kind of need to understand the basic anatomy of a tubular or sac-like organ, because so many of the organs we're going to look at are either tubular or they're sac-like. So a tubular organ would be something like a vein or artery, a capillary, the vagina, the small intestine and large intestine. Okay? These are tubular. And the other organs are sac-like, meaning that instead of looking exactly like a tube, they look more like a bag, like the stomach or the bladder or the uterus or the heart or the gallbladder. But there's lots of organs like this. Now, when you look at a tube-like structure like this, let me just show you a tube that I managed to find when we were taking a break. Okay, here's a tube. It doesn't make any difference if it's backwards or forwards. This is whether it's in reverse or not. Okay, this is just a cardboard tube. When you have a tube like this, and of course I'm thinking about epithelium, when you have a tube like this, there's sort of two parts to it, right? There's this part here that I'm grasping with my fingers, right? It's this part right here. Whoops. Yeah, this part right here. I guess I'm touching it when I'm out here, right? I'm touching it out here, okay? And this whole portion is called the wall of the tube. This is the entire wall right here. I'm pinching it, right? But this is also the wall on the outside. Now, if you have an actual tube, then it has a hole through it like this, an empty cavity inside of it. And if I hold this just right, you can hold this just right and try to make it look. I'm trying to make it look like one of those tubes we were just seeing. Maybe it's not going to work that way. So we're seeing the edge of the tube here. This empty space inside the tube, empty space in here, this empty space is called the lumen, L-U-M-E-N. And lumen actually means light. So it's kind of like when you look at these slides, you see light through it, right? And that's why they early histologists just decided to call that the lumen, or light area where it's empty space. Whenever you find a lumen in the body, you will find epithelial tissue lining that lumen. So every one of these organs has epithelial tissue lining the lumen. So here's the wall. Okay, this is also the wall here, cross section. There's connective tissue out here that forms a sort of capsule that supports everything. Then there are multiple layers of cells here. Here's the lumen, or empty space. This was the small intestine. This would be full of food. If this was the urethra, this would be full of, could be full of urine. Now, when we analyze this wall here, we see connective tissue, muscular tissue, connective tissue, and epithelial tissue. The epithelial tissue always lines the tube like this. Between the lining of the tube, where it's epithelium, and the wall of the tube, there's this layer, red here, of smooth muscle. And some tubes have a lot of smooth muscle. In fact, almost all of the tubes that are listed up here have smooth muscle. The only one I can think of that does not is capillaries, because capillaries are so thin. And actually, veins may not have much smooth muscle in it. But we've got empty space, the cells that are in blue that line it, the epithelial lining, connective tissue, smooth muscle tissue that can contract or relax to open and close this tube, and then the wall of connective tissue too. So understanding what a tube or sac-like organ looks like is important in understanding how epithelium is placed into an organ like that. So there are covering and lining epithelium, like the epidermis of the skin. There's epithelium that lines blood vessels, that lines hollow organs, like I just mentioned, that forms the inner part of most membranes. There's also glandular epithelium, the kinds of epithelium that lives in endocrine or exocrine glands that is involved in secretion. Gland sounds like secretion to me. So secretory cells are part of all glands. They line the ducts of glands. Remember exocrine and endocrine glands. How could you forget exocrine and endocrine glands? Now, you've heard these names for epithelium probably, like ones like this. Non-keratinized stratified squamous epithelium, or simple cuboidal epithelium, or pseudostratified columnar epithelium. Believe it or not, in a few minutes, you're going to understand what those names mean. Because whenever you name a connective tissue, you always give at least 2 bits of information about it, like simple cuboidal. And sometimes you even give more than that. But the very first thing you usually hear about a tissue has to do with how many layers thick it is. If it is a simple epithelium, it is 1 layer thick, 1 cell thick. If it is stratified, then it is 2 or more layers thick. And honestly, there are parts of the body where we have stratified epithelia that's 30 or more cells thick. Okay, so first is simple, like simple cuboidal, simple columnar, simple squamous, stratified squamous, stratified cuboidal. So it refers to the number of cell layers. Also included in the names all the time is the shape of the surface cells or the cells, right? And the cell names are these. There's what are called squamous cells that are thin and flat, cuboid cells that are round and shrimp-shaped, and columnar cells, which are long and thin, like columns. So now you've heard it. Simple cuboidal epithelium means it is 1 layer rounded tubal cells. Or if I said stratified, it was many layers of particular cell, but the surface cells, because that's what apical means here, so cell layers, if it's say stratified squamous, it means there's many layers of cells, and the cells in flesh. Maybe some other modifications like cilia, microvilli, or keratin. Like I have already said, the name for the epidermis of our skin, that is the tissue that we mostly see when we look at somebody. It is called keratinized, stratified squamous epithelium. All right. So, keratinized stratified squamous. Take a look at it. Well, we know it's epithelium because honestly, we wouldn't have heard stratified or squamous if it was any other kind of tissue. Stratified means many layers thick. Squamous means the surface cells are thin and flat. And keratinized means that the surface cells are full of keratin. All right, so just, for the record, that gives you a specific example of what you are likely, the kind of names that we generally hear when we talk about epithelium. So I am modifying this, so I will repost it. So for what it's worth, we're going to find epithelial tissues all over the body. We're going to find them lining the esophagus. Oh, now take a look. Non-keratinized stratified squamous. That's what we find in the mouth, in the esophagus. Non-keratinized means the surface cells are alive with nuclei, no keratin on them. Simple squamous epithelium, it forms the air sacs of the lungs. I mean, I know it says lining the air sacs of the lungs, but it makes up the air sacs of the lungs. Simple cuboidal epithelium, we saw this before, forming a tube in the kidney. Simple columnar epithelium, lining the intestine. Keratinized stratified squamous epithelium, human skin cells. The surface cells are dead, flat layers. These are the things that are always flaking off. So let's talk about simple epithelium. And there's three kinds: simple squamous, simple cuboidal, simple columnar. Let's talk about simple squamous epithelium first, right? Simple squamous means one layer of thin and flat cells. It's a single layer of thin, flat cells. It can sort of look like fried eggs in that can be so thin that the nucleus kind of bulges up into the lumen and is visible. These cells are tightly packed together with very little intercellular space. Their function is to be thin, flat cells, ideal for exchange. These are cells that are very commonly positioned in the body for diffusion, osmosis, and filtration. I mean, there also can be found in protected areas where slick, slippery surfaces are needed to reduce friction, like where they line blood vessels and the heart. And in fact, when they line blood vessels in the heart, and the blood, where they line blood vessels and they line the heart, they are referred to as endothelium. I mean, they can just as accurately be referred to as simple squamous epithelium. But very commonly, when you find these lining the heart or lining blood vessels, they are going to be referred to as endothelium. Okay, so just for the record, it could be called endothelium. Where are you going to find simple squamous epithelium? Well, you find it lining blood and lymph vessels and the heart. Find it lining cavities in the body. You find it forming the respiratory membrane, which is the air sacs of the lungs plus the capillaries. You find it in the kidneys, where it's involved in filtration, reabsorption, secretion. It is the tissue that forms the tubes where blood is filtered and cleaned up by the kidneys. I mean, this is a surface view of it right here. And can you see how the nucleus, these cells are so thin, the nucleus is kind of bulging up and makes it look. Now, remember, this is simple squamous epithelium. So this is just going to be one cell layer thick. You find this forming the actual walls of capillaries. This is a capillary here. Remember I showed you a picture of a capillary and I said red blood cells have to march through capillary pretty much single file? Well, this capillary is actually formed from epithelium. And some of the epithelial cells that form it are literally so thin and flat, they form a tube that way. I will show you in a second what this looks like. Okay, here's the cells. This is another blood vessel. This is smooth muscle, and this is the lumen here, and here's the cells that are lining the lumen. So one place we see epithelial cells is lining the heart, lining blood vessels. Again, these are blood vessels here. So here's a cross section through a blood vessel. See these nuclei? These nuclei are all simple squamous epithelium. In the lungs, my gosh, you look at the lung tissue and you pretty much see simple squamous epithelium and alveoli, and that's, forming the air sacs of the lungs. Here's the air sacs of the lungs right here. This is the air sac, and here's the cells lining it, which are called type 1 pneumocytes, type 2 pneumocytes, but that type of tissue they are is epithelial tissue. Capillaries that surround the blood vessels of the heart are made of simple squamous epithelium. Now, before we leave this, let me just show you, just illustrate something about the way these cells are put together. I wish I had my model. I built a great model of a capillary, and guess where it is? It's locked in my classroom, and there's no way they're going to allow me to get anywhere near it. They're talking about letting people come onto campus next week to do some filming of labby type stuff starting next week, but they're not going to let us onto West Campus. It's only campus they're doing that filming and stuff on is Northwest Campus. So anyway, let's get aside. Here's one, my hand, pretending to be one simple squamous epithelial cell. And here's the other hand pretending to be a simple squamous epithelial cell. If you knit these two together, look what you form, a tube. And at the capillary level, some of the smallest capillaries are literally 2 cells glued together like this with an empty cavity or lumen inside of them, okay? And they are made of only 2 simple squamous epithelial cells held together by these tight junctions that knit the cell membranes together. And they form a very simple tube that is a blood vessel, a capillary, and that blood cells will somehow sneak through. So simple squamous epithelium. But you know what? Sorry, I forgot to show you this. I have posted on my PIMA a couple of documents that I'd like to make available to you. They are things that I put together, and I want to show you the first one. Well, there's 2 of them, right? So where did I put these? Now, these are posted, did I say on my PIMA? I apologize. These are posted on D2L in the content line. And what they are called is histology images for study. And they look like this. Now, what I'd like you to see about is I'd like you to see that what I have done is taken all the major tissue types that we should be discussing, and I have summarized information about them and included some kind of image, which is usually, I think, a pretty good image. So this one is called Histology Images for Study, E&N, which stands for epithelium and nervous. And take a look at this first image, image 1, epithelium, simple squamous. So here's which tissue type it is, epithelium. Here's what its specific name is, simple squamous epithelium. When it lines a blood vessel or the heart, it's called endothelium. Here's its function, to form capillaries and air sacs and promote diffusion. And locations include lining blood vessels, air sacs of the lungs, the kidney capsule, etc. So what I've tried to do is summarize some of the most important basic information, and I put it together in a document that I'm handing to you on a silver platter. Feel free, make a copy, use it in Bio 201 or whatever. Now, we looked at simple squamous. You know all about it now. Here's the next one, simple cuboidal. Simple cuboidal, you could probably guess. Simple means 1 layer of cells. Cuboidal means that the cells are sort of cube-shaped or roundish. You know, this is a cross-section through the kidney again. This is one of those tubes that helps your body create urine. And notice that these cells form the walls of this tube. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 cells that are knitted together to form this. And then this at the bottom of those cells is the basement membrane. So this layer right here is the basement membrane. And then here's an individual cell. Is it cube-shaped? I don't know. It's roundish or cube-shaped. When you see what a simple cell looks, simple squamous cell looks like, really thin and flat, well, this doesn't look like that. Simple columnar is going to be longer and thinner. One thing about simple cuboidal cells is they often have really nice, conspicuous, round nuclei. Nice, conspicuous, round nuclei. Round nuclei. What's this white space called here? I'll give you a hint. Oh, what did I do? I didn't mean to do that. Well, I'm going to keep this document up because there are some really cool images here that I want to There we go. Anyway, there are some really cool images here that I want to utilize, so I'm going to reduce this down to a reasonable size. And I'm going to keep this document live down here so we can look at it. And you have a copy of it. I provided for you a copy of it. All right, simple cuboidal. Well, simple means one letter, cuboidal means cube-shaped cells. These cells look a lot like what I just showed you. There's the basement membrane. There's the connective tissue. That's where blood vessels are going to be found. Single layer of cube-shaped cells, round nucleus in the center of the cell, very little intercellular space. Ooh, sometimes they have microvilli. Kidney tubules especially, because kidney tubules are one of those places where a lot of water absorption needs to happen. And the more, if you really need to absorb stuff, then you're going to have the surface area of your cell increased by microvilli. Now, these cube-shaped cells deal for absorption and secretion. They can be a barrier in protected places. They can be involved in transport. Okay, I honestly think that what's missing from here and that needs to be put in is this. Can be used to form tubes. And when we look, well, some of the tubes we've already looked at, we can see they're being used to form tubes. Okay. So, like I said, it can be used to form tubes. I mean, take a look at this tube here. This tube is formed by them attached together. The tube-shaped cells ideal for absorption and secretion, like in the kidney tubules. They can be a barrier in protected places. They can be involved in transporting, meaning they form the tubes that transport substances. Oh, here's a tube, again, formed by simple cuboidal epithelium. The kidney tubules, well, do you recognize this picture on the right? This picture on the right is one that I've used multiple times. It's in the document I presented. Here's the lumen. Here's the cube-shaped cells. Here's the basement membrane. Some of this stuff is kidney tubules, like here's another tube and here's another tube. And some of this stuff, these things that you see here and here, these are blood vessels. So what's happening here is water in the middle of this is being moved by osmosis through these cells and into blood vessels. I mean, your kidneys are so efficient. They filter something like 35 times your total blood volume in 24 hours, which means if you didn't reclaim about 99% of that water, you would urinate yourself to death in an hour, right? Because there's so much water lost. But instead, in the kidneys, most of that water is reclaimed, and most water reclamation happens thanks to simple cuboidal epithelial cells, which take the water, bring it into their bodies, and then pass it to blood vessels. Right, next one is simple columnar epithelium. Now take a look at these cells, long and thin. Ooh, a goblet cell is a type of simple columnar epithelium. So I would say simple columnar epithelium is a specialized type of cell. But then within that specialized type of cells, the goblet cells that are even more specialized. What do goblet cells do? They make mucus for protection. Mucus is a protective secretion. They often have a prominent oval nucleus close to the bottom of the cell, though it can be in different places. These are highly adapted for absorption, secretion, the movement of particles. Again, they form the lining of tubes like the intestine. The lining of the intestine is a good place to find them. So some structures that can be found in simple columnar epithelium are microvilli, increase the surface area for better absorption. Cilia, sometimes simple columnar epithelium can be ciliated. Like when I think of ciliated simple columnar epithelium, I think about the oviduct, which is something we talked about earlier today. The fallopian tube or the oviduct, the tube that will carry an egg from the ovary to the uterus. It's called the oviduct, or it is called the fallopian tube. They can have goblet cells for the secretion of mucus, and many do. So in this particular picture, here's the simple columnar epithelium here. Can you see the nuclei have all lined up nice and neat? And then stuck in them, look at this, these weird cells here, big blobby weird cells. These cells are goblet cells. Where you find simple columnar epithelium? Lining the stomach, lining the intestines, lining the gallbladder, the bile ducts, lining the uterus, lining the fallopian tubes where they are ciliated, lining the ducts of the testis, meaning the ducts that carry sperm out of the testes and towards the outside world. You can find them lining secretory ducts of many glands. You find them all over the place. Oh my gosh, they line the intestinal macrovilli. So these fingers here, sort of sticking up here, this is a finger. This is called a macrovillus, or just a villus. It is a finger-like projection in the intestine. Now, when we see this, I automatically go, this white stuff here is not cellular. That must be the lumen. And so what cells always line a lumen? Well, the cells that always line a lumen are these cells, which are called simple columnar epithelial cells. But even within the simple columnar epithelial cells, take a look. Goblet cell, goblet cell, goblet cell, goblet cell, goblet cell, goblet cell, lots of goblet cells. And those goblet cells, of course, are going to make mucus to protect the wall of the intestine, really. You find them lining. So let me just go back to this for a minute. This thing is like a finger-like projection that sticks up off the wall of the intestines. The intestines are pretty amazing. They're folded like this. Then sticking up off those folds are macroscopic fingers. That's why they call them macrovilli. Villus means finger. So they stick up off. And these are big enough to see. They kind of look like shag carpet or velvet if you look at the lining of the intestine. But then these, of course, as you saw, are covered with simple columnar epithelial cells. And those simple columnar epithelial cells, those simple columnar epithelial cells, they have microvilli on them. So this is the simple columnar epithelial. Well, this is a goblet cell, goblet cell, goblet cell. This is a simple columnar epithelial cell. This stuff that you see right here, which is kind of hard to see, but it's magnified here, is microvilli. So I don't know how many times I've read this. When you add up, your small intestines may be about 10 feet long in your body, about 10 feet long. It's folded inside. It's got macrovilli sticking up. Those have simple columnar epithelial cells on them, and all those simple columnar epithelial cells have microvilli, okay? This makes for a huge surface area in the intestine. Why does a small intestine need a huge surface area? One word, starts with an A, absorption. That's one of the places where digestion and absorption are so important. Now, between the folds and the microvilli, the surface area of the small intestine is approximately the same size as the surface area of a standard tennis court. And that is a lot of surface area for absorption, no doubt about it. Nice lined up nuclei. Here's the lumen. This stuff down here is connective tissue. Some other images. Big plump goblet cell. This is microvilli here. Notice the nuclei are kind of lined up. Nice and straight like that. Now, we need to talk about a weird tissue called pseudostratified columnar epithelium. And it's a simple columnar epithelium, meaning it's a single cell layer. All the cells are attached to the basement membrane. Not all cells reach the apical or free surface. Nuclei can be at different levels here. And here's the reason why it has such a weird name. Pseudo stratified. Well, we know what columnar means, column-shaped, thin and tall and thin cells. Pseudo stratified, well, pseudo means false, and stratified means layered. It is falsely layered. And what that means is that some of the early histologists who look at this, they were basing their decisions about whether it was layered or not on whether they could see different layers of nuclei. And when some of the early histologists looked at this, they thought, oh, wow, this is stratified. Meaning there's more than one cell layer. But that really wasn't true. Let me show you a really good image here. It's one part of this packet that I put together for you. Down here is the blurb about simple columnar epithelium. And this is simple, remember. Notice the nuclei pretty much all in a nice neat row. Goblet cell, microvilli. Out here is the lumen. This is actually kind of cool, really. One of the reasons I like this image so much is because this is the lumen here, right, of the intestine where food would be. Here's the cells that line the intestine. And take a look. This is a goblet cell back here that is actually oozing out mucus. They caught this perfectly when they were making this slide. Now, notice that this is clearly simple, nice, thin layer of nuclei. What I want to compare it to, I may need to shrink the image a little bit to do this. What I wanted to compare it to, no, I definitely did not mean to do that. Okay, here we go. This is perfect. I wanted to compare what this layer looks like to what this layer looks like. Because here, nice, evenly layered nuclei. Over here, this is pseudo-stratified columnar, but look at the nuclei. It looks like there's nuclei way down here, nuclei kind of in the middle, nuclei at the top. There's a goblet cell. There's some cilia on the surface of these cells. But the bottom line is that look at how the nuclei are all over the place. It almost looks like there's like 1, 2, 3 layers of nuclei, which is why I think early histologists thought this might be stratified. But you can take this tissue and dissect it carefully and discovered that there's just one layer of cells. Hence the name pseudo-stratified, falsely stratified. And so you will find this. We're going to go back to the PowerPoint in a second now. Okay. Leave this up here. We're going to go back to the PowerPoint if I can find it. There it is. Single cell layer. Looks stratified, but it ain't stratified. Nuclei at varying depths, the function, absorption, secretion, movement of particles. I mean, these cilia here are clearly there to move things around. You know, when I think of pseudostratified columnar epithelium, I tend to think that you're going to find it in the respiratory tract, where its job is to move particles around. I mean, every time we inhale millions of little fine particles. We don't want to let those things settle in our lungs. You know, I can remember back to being in, I think it was 4th grade or 5th grade. And one day, out of the blue, they came and they took your class away. They sent all the boys to one room and all the girls to another room to take what they called a hygiene class. And the reason they separated the boys and girls, I think, was because they were going to talk a little bit about sex and make you like look at a cross section through the pelvis and label parts. And they were afraid of, look, you can't leave a bunch of 5th graders in a classroom, in a co-ed classroom, and talk about sex, or otherwise you're going to have some kind of 5th grade orgy and have a bunch of parental complaints. So they sent all the boys to one room and all the girls to the other room. But one of the things I remember so well was there was this white-haired woman there who waved her finger at us and said, listen, you should better never smoke cigarettes because you will burn the little hairs off the linings of your lungs. And everybody went, oh, gross, hair in our lungs. But what she was, of course, referring to was the cilia that were there, which can be paralyzed or damaged by cigarette smoke, other kinds of smoke. So she was basically warning us against that. But she called these things little hairs. I suspect she knew that they were called cilia. But whenever I think about pseudostratified columnar epithelium, I think about that woman and her warning us not to smoke because of the little hairs. You find it in the upper respiratory tract. You can find it in the epididymis and vas deferens. And if you don't know what those are, the epididymis and vas deferens, okay. These are tubes transport Now, why would you need cilia in these? And I mean, that is a good question. Why would you need cilia? But you probably know the answer because you remember that I said earlier, look, sperm don't get to swim inside the male body. They're paralyzed. So if you're going to move sperm gently and respectfully out of the testis and up into the tubes that carry sperm and semen out of the body, you're going to need cilia to help with that. You know, when you have a vasectomy, you will cut through the vas deferens, cut it, burn the edges to seal each other, and all of a sudden now sperm are stuck in your testis and they're never going to leave there. Okay. You can find this in the male urethra, which seems odd to me. So here's another image of ciliated pseudostratified columnar epithelium. Notice there's a layer of nuclei down here and a layer of nuclei up here. That's what made it confusing. Here's some beautiful cilia. What a great image. And here's some goblet cells making mucus and secreting them out. So you could inhale particles. Those particles could get stuck in the mucus that is in your respiratory tract. And then the cilia can move that particle-laden mucus into a position where you can swallow it or cough it out or sneeze it out or whatever. cilia here. I don't think we need. So classifying epithelial tissue, number of cell layers, shape of apical cells, modifications of apical cells. Okay. Now let's talk about stratified epithelium. Stratified epithelium is epithelium with layers. So when we talk about stratified squamous epithelium, we get to talk about some of the most interesting tissues of the body. Like when you have stratified squamous, well, let's just think about this for a minute. Stratified means multiple layers. Squamous means thin and flat cells on the surface. Well, of course, epithelium. Now, when we talk about stratified squamous, we're going to be distinguishing between keratinized stratified squamous, okay? Keratinized stratified squamous. And non-keratinized stratified squamous. Okay. Now, so stratified squamous, this is mostly what we're going to focus on. But there is stratified cuboidal. There is stratified columnar. And then there's another weird kind that it changes back and forth from stratified to stratified during the day. Now, stratified cuboidal and stratified columnar are relatively rare tissues. We're not going to spend much time, if any time, talking about those. I want to talk about stratified squamous. The first one, keratinized stratified squamous. That means that the surface cells are thin and flat, but they're the surface cells in a bunch of layers of cells. And lastly, these surface cells in a keratinized stratified squamous are going to be filled with keratin. Now notice down here, do you see this layer here? That is the sternum germinativum. This is the basement membrane right here. And this is connective tissue with a blood vessel there to show you that. Look, blood vessels are pretty close to these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 cells thick. So not a bad picture of it. Several apical cells are squamous cells. Surface cells are dead and filled with keratin. Keratinized stratified squamous epithelium, really protection, a barrier. Where are we going to find it? In the epidermis of the skin, the outermost tough layer of the skin. Right? The epidermis means on top of the dermis, and so it's the outer layer of skin. If you look at somebody, this is what you see. You see keratinized stratified squamous epithelium. Some parts of the body have a pretty thin layer, like the lips, which are definitely part of the skin, have a pretty thin layer of keratin, which is a good thing. It allows you to taste your food better and feel kisses better and all that kind of stuff. The keratin layer can interfere a little bit with sensation because it's layers of dead black cells. You can find it in parts of the tongue. So actually, take a look over here. This is the keratinized stratified squamous epithelium. The bottom, the stratum germinativum, is right down here. And all the way from there up to here, the top of the stratum spinosum is, these are all living cells that are stratified. We can't really count the cells here. But at a certain point in time, the cells at the very surface die, and those cells have turned themselves into keratin, And that keratin gets all compacted, so we have a pretty thick keratin layer. Now, sometimes that just looks like this, right? Because here's the stratified squamous. Let me show you one of my favorite images, stratified squamous. Here's a good one right here, okay? Starting here, this is the stratum germinativum, and this is the stratum germinativum down here. These are layers and layers of cells. This is the lumen, which means the outside of the body. And this layer of flaky stuff, that is definitely keratin. Now, the other image I have here for you is this one. Okay, now again, this area right here is the epithelial cells, starting with the stratum germinativum, and they go all the way up to where they start to produce keratin. And look at this gigantic, hundreds of layers of dead, flat cells. This is like the very thick keratin that's on the bottom of your feet. Very thick keratin on the bottom of your feet. This other image here is for much thinner layer of keratin, maybe the keratin on the surface of your face or your arm. I mean, again, you know, there's places where the keratin is thicker, like on the bottom of my feet, because I'm stomping around on my feet all day, compressing those keratin layers. And then there's areas like the face and the lips that the keratin layer is thinner. And of course, you can even find a type of stratified squamous epithelium that does not have keratin on it. And this is one of my favorite images of that. Take a look. This is non-keratinized stratified squamous epithelium. And honestly, whenever I look at an image like this, I sort of think, can I find the lumen? Oh, well, here it is right here. Here's the lumen right here. This is the lumen, this thing right here. This is the empty cavity. Now, look, I got to tell you, this cross-section here, it is either a cross-section of the mouth, it's a cross-section of the esophagus, or it's cross-section of the vagina. It's very difficult to tell. I actually think this is the vagina, but really the lining of our mouth, the lining of the upper part of our esophagus, and the lining of our vagina, if you have a vagina, I don't, then the lining is non-keratinized stratified squamous epithelium, and it looks the same. Now, let me fill you in here. This dark line right here, these are the stratum germinativum cells. These are the mother cells for this entire layer. And they are going to divide a couple times a day. They'll go through the entire cell cycle a couple times a day, and they will produce many, many layers of cells. So I can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 33 layers of cells. The function of tissues like this is absolutely protection. I mean, these are the cells that you scrape them off when you brush your teeth or you eat some nachos. They tend to be in kind of wear and tear areas of the body where there are delicate tissues that need to be protected by many layers of cells. And these surface cells here, they can easily fall off. Let me show you a couple of electron micrographs, a couple of photomicrographs I'd like to show you. Oh, please, I hope I can find these really quickly. Hmm. All right, just give me a second. I can't find these in 30 seconds. I'm gonna. Hmm. Hmm. Cool pictures here, but not the ones I'm looking for. All right, like I said, if I can't find these in a few minutes, I'm going to give up. I really wanted to show you a very interesting set of images and where they are. I thought I had them right here. Well, these are vaginal lining cells, but they're not the exact images I was looking for. Darn it. Folks, that was a waste of time diversion, and I need to find those. Sorry. But there were some images I had taken of cross-section through the vagina where you could just literally see these cells falling off because that's their job, to fall off and protect. Okay. Oh, how about non-keratinized stratified squamous? That's what we're just talking about right now. Many cell layers. The top cell layer of cells is flat, but this is interesting. The cells on the top of a non-keratinized stratified squamous epithelium are alive. They are living cells. Now, again, take a look at this one image I do have up here. And see how there are nuclei right here on the surface? Well, that says that these are living cells. And in fact, if these cells, as they flake off, they are going to be living cells. Now, when you look at keratinized stratified, like this one, this flaky stuff that's on the surface here, this is dead, flat, keratinized cells. There's no living cells on the surface. If you were to scrape some of the surface of your epidermis off and look at under a microscope, you're going to see no living cells, right? But if you were to take a sample off the lining of your mouth, you would definitely see living cells. I mean, the bottom line is that these are some of the images I was trying to find a minute ago, but I'm going to go right here and put what happened to it. That was a nice one. I wanted to look at that one. Oh, there it is. These are cells that were basically scraped right off the lining of the mouth. And of course, this is going to be just as difficult as possible. These cells here, this is a mouth lining cell. Take a look. Here's the nucleus. These are living cells. This is when you see a pap smear, which is the lining of the vagina that's been the cervix and the vagina, that has been a very careful sample of that. Well, I want to see micro. I want to see the actual cells. Gosh, I don't see all this stuff. What I want to see is what the cells look like. The pap smear, here we go. Well, of course, I'm going to try and make it as difficult as possible. But what I wanted you to see was look at these cells. These look just like mouth lining cells. Why? Because they're the same exact kind of cell lining the vagina that line the mouth. And when you look at a pap smear, you will always see some of these cells that are just like mouth lining cells. I apologize for some of the imagery there. I hope I didn't offend anybody. I was looking for cells and managed to see other stuff. But anyway, okay, back to here from current slide. Where do we find it? In moist areas that need lots of protection, sometimes wear and tear areas, okay? And you're going to find it in the mouth, parts of the tongue, in the throat, in the esophagus, in the vagina, in the anus. You're going to find these many layers of cells as protective cells. Again, what's cool about this image is you can see up here at the surface, I see nuclei right at the surface. Here's all these layers of cells. Here's the stratum germinativum down here. This is the connective tissue, or the dermis, of this kind of skin. I mean, we don't often call this skin, but it's lining. It is a type of skin that has the same exact structure as normal skin. Here's kind of a cool idea, or a good, cool idea I'd like to show you is when you think about your face, this skin right here, keratinized stratified squamous epithelium, it's probably not as thick as the keratin that's on the palm of my hand or my elbow, or certainly in the bottom of my feet, but it's keratinized. Now, as you move towards your mouth, you start to touch your lips. The keratin gets very thin, and that means the sensory neurons that are near the surface are going to be more sensitive there. But then you go inside your mouth, and when you're inside your mouth, what you're touching there is non-keratinized stratified squamous. Keratinized stratified squamous, very thin keratinized, no keratin inside your mouth. But the structure of that is pretty much, it's identical to the basic underlying structure. The question is it got keratin on the surface, these many, many layers of dead cells, or not? And we find them. Oh, hey, here's one of my images. I forgot it. There's a great image there. This is a cross-section through the vagina, and take a look at these cells on the surface that are flaking off. We see them at this angle on the side. We see them over here. Stratified squamous epithelium of the tongue, stratified squamous epithelium of the esophagus, vagina. These cells look like this. Now, the last one I want to talk about is transitional epithelium. And transitional epithelium is a kind of epithelium that is stretchy. It is multi-layered, so it's appropriate to talk about it here when we're talking about stratified. But here's the thing about it. It can change the shape, and the shape of the cells can actually change from 1 minute to the next. The most common place to find cells like this are in the urinary bladder. And that's because the urinary bladder has to expand and contract multiple times a day. When your bladder is empty, the cells are very cuboidal. But when you start to stretch the bladder and fill it with urine, it stretches this stretchy tissue, and the surface cells go from being very cuboidal to being more like squamous cells. So they actually change their shape. I mean, take a look. Here's a nice round cuboidal cell. This cell looks more squamous. And, you know, I've had a bladder my whole life, and I know this. It can be really full 1 minute and all stretched out, and it looks like this. And then 1 minute later, it can be empty and you know, contracted and smaller, and then look like this. So this word transitional, it means changeable, right? Transitions means change. And so this is a type of epithelial tissue that can stretch, and the shape of the cells will actually change multiple times during your day because of this stretching. Now, we've talked about glandular epithelium a little bit already, endocrine glands, exocrine glands, so I'm going to not worry about that. Oh, goblet cells, we've again talked about goblet cells many times. This is old, and there we go. Epithelium in a nutshell. I mean, really, that's probably not true. I don't think there's epithelial tissue in nuts, but you know what I meant. Epithelial tissue in a nutshell. Now, I'm going to repost that because I made some little tweaks in it, and I want you to have the tweaky version, not the tweak-free version. Okay. I don't know about you, but I'm exhausted. I mean, you're exhausted from listening to me. I'm exhausted from talking. Take a look when you get a chance, though, at this wonderful document that I provided for you. And this one has epithelial tissue, and it has nervous tissue at the bottom. So I'm actually going to post the PowerPoint that deals with muscular tissue so that you can take a look at it before Monday, and nervous tissue so you can take a look at it before Monday. And take a look at this document too and study it at great length. The other document is called, I'll show it to you. The other document is right here. And that one, remember, was called Histology Images for Study. That one was called Histology Images for Study, ENN for Epithelium and Nervous. This is up there too. Histology Images for Study, CNM, which of course stands for connective and muscular tissue. And the top of it is all connective tissue, which we will certainly talk on Monday. And the bottom is muscular. Okay, muscular tissue. I'm also going to post a document that compares the 3 types of muscle tissue from a cellular perspective. I'm going to post that before I am done on the computer today. So this image, this is already published for you to look at. I'm going to show you a document that I created that summarizes the 3 types of muscle tissue. I'm sorry I haven't had a chance to talk about this yet, but I'll post this.
Wait, is this on D2L?
Yes. I'm not
Okay,
posting anything on my Pima anymore because people are having trouble getting to my Pima. This isn't on D2L yet, but I am going to post it on D2L as soon as we're done. Now, in addition to that, of course, I will also post the PowerPoint that deals with muscular tissue. Okay.
And what PowerPoint is that under?
It's going to be, well, the PowerPoint is this one here. It's called muscle and nerve tissue. I may,
Okay.
you know what? I may have already posted this. Anybody
Okay,
see? I already posted this one on muscle and nervous tissue. Anyway, it's going to be up there in a few minutes. Okay. I'm going to go
Okay.
double check. If it's not up there, I'm going to put it up there so that people can study over the weekend. I know we got a lot of stuff to do, like the exam and all that, but if you would look at these PowerPoints, it would be good.
Okay, thank you.
Very useful information. Welcome. Any other questions or concerns? Thursday class is over, I think. I can't do anything else. I am burnt out. So I'm going to stop recording.